So, very good morning to everyone. So, how are all of you? All going well? Am I audible? Visual? Voice is still not audible? No voice? Now it is okay. Right, very good. Now it is audible. Good, good, good. Very good. So, yesterday respiration not completed. Let's see. First, me try to complete what are the schedule of the day. If we find any time, we will try our best to finish whatever the pending is there, right? So the very first agenda is entire second year botany, as well as from the first year morphology and anatomy. If time will permit us, allow us, we will take care of it, right? So what we are planning, like, what happened? We are just going to talk about the some of the area where you are missing first. Means I am not starting with morphology. I am not starting with microbe. I know you people better know about it. So I am starting with the biotechnology first. A reason? Because weightage is also good, and somewhere some people get stuck also. So the entire effort is to talk on the hits area. So from where probable questions is going to ask. So listen it carefully, patiently. It is a marathon for me also and as well as you also. To listening a teacher continuously 5 hours, it is also not an easy job. So maintain your patience and let's start. Okay, right. So we are starting with biotechnology. First chapter, then we will talk about the biotech. Second chapter, then we will talk about the molecular basis of inheritance, then principle of inheritance, then reproduction in such a way, and if after that, of course, microbes in such a way we will take care about which one? Second year topic, then morpho and in our first year, okay? So these are the one student is saying, sir, one doubt is there. So if it is related to this, you ask the doubt, otherwise we don't have today that much time to address the personal doubt here, right? So you just WhatsApp me your doubt if it any questions is there. If it is related to marathon, you can just ask your doubts, right? Not a problem. So let's start now. So we are starting with very first chapter of biotechnology, right? So let's start with the introductory. You just please open your NCERT because what I find probably the textbook not visible very properly in your mobile. If some of the students are watching in mobile, I also watched it, I observed it. Visibility of the textbook line is not very clear. So you have studied already the NCERT many times. So you know about which line and which paragraph I am saying. And you please open your textbook as well. Got the idea? So we are starting with the definitions of the biotechnology. Just we will try to cover almost area, right? And at least who is highly important. So we are talking with the definition. So what NCERT is writing? Biotechnology deals with technique of using live organism or enzyme from organism for product and processes useful for human being or useful to human being. So what they are telling, in biotechnology, we are either using the live organism, living organism, or we are using the enzymes from the living organism for the product and processes which is useful for human being. Means entire approach of the biotechnology is what? Anthropocentric, by keeping human welfare in the mind. So we are using other organism for human welfare. In literal meaning, you can think like this. So in base of that, NCERT wrote what? Key biotechnology, if you talk about making curd, making alcohol by fermentation, they also comes under the biotechnology. Because there also, you just try to understand the curd making. In curd making, we are adding which one? Lactobacillus in milk. It is converted into curd. What we are using? Living organism we are using for making the product of our choice. So we are exploiting that living organism for our services. Are you getting the idea? So this is biotechnology. But that is a old biotechnology. Now what is new biotechnology? What is the latest biotechnology? In new biotechnology, we talk about genetic modification. So whenever we are doing genetic modification, means living organism have no capability. Suppose that there is a bacteria. Bacteria don't know how to make insulin because they don't have gene. But what we are adding, we are taking a human gene and incorporating them. So we make them transgenic, we make them genetically modified. So when we are using the genetically modified organism for product and services of human wing, it is called what? Modern biotechnology. So biotechnology in modern sense using which one? Genetic modified organism and transgenic organism. Are you getting the idea? Classical biotechnology, old biotechnology is alcohol making by process of fermentation, yeast we are using. Making of, you can say about which one? Curd, it also comes under the old biotechnology. Now what NCERT say now? So nowadays, who is also included under the biotechnology? Test tube baby, in vitro fertilization also nowadays is a part of which one? Biotechnology. Are you getting the idea? These are the line which is usually going to ask. I am telling here. Test tube baby is also a part of which one? Biotechnology. Getting the idea? In vitro fertilization. That definition is not that much important. What is the definition of the biotechnology given by European Federation? Not that much important, so I am not addressing about the definition. What the definition? They told, simply see in one line. So they told about biotechnology. The European Federation of Biotechnology has given definition of biotechnology that encompasses both traditional view and modern view. And what they told? Integration of natural science and organisms, their cell, their part, and their molecular analog for product and services for the human being is called biotechnology. This is the definition given by which one? European Federation. Definitely it. Now we are going to move on the next area. Principles of biotechnology. Many time in paper B ask also, examiner ask also, what are the two key principles involved in biotechnology? One is genetic engineering. Another principle written in next page is which one? Bioprocess engineering. Bioprocess engineering. So what we do in genetic engineering? In genetic engineering, we are developing a genetically modified organism. We are developing a transgenic organism. We are developing a chimeric organism. So genetic engineering is also called what? rDNA technology. It is also called rDNA technology. Got the idea? So what we are making in genetic engineering? Genetically modified organism or transgenic organism or chimeric organism. Are you getting the idea? And other name of genetic engineering is what? rDNA technology. Understand? Clear or not? So how many component of two main principles of the biotechnology? Two major component is, one is genetic engineering, another is bioprocess engineering. What we are doing in bioprocess engineering? We are culturing the recombinant host cell in a contamination free environment in all ideal condition. What is it clear? So what happened in bioprocess engineering? Suppose that we are taking a bacteria who have insulin producing gene. We are growing in the culture medium. We are growing in the culture medium. In culture medium, what happened? But bacteria number will multiply. Our desired gene is also multiply there. Understanding the idea? So multiple copy of the desired gene will produce. Gene cloning occur. And in the host cell, everyone will express and produce the desired product. So this desired product may be hormone. This desired product may be antibiotic. Got the idea? So that we perform in bioprocess engineering. So we are making genetic modification via this process, genetic engineering. We are culturing and making large number of the host cell. So in every host cell, desired gene will express and produce the product in large amount. We are doing in bioprocess engineering. So don't forget these two things. These are the two main principles of the which one? Biotechnology. Now we are moving ahead. Now we are moving on the next page. Bioprocess engineering we already discussed. Now in this paragraph what NCERT is writing, I am just telling you. So what we are doing in the biotechnology, just try to understand. Suppose that this is a host cell. This is a bacterial cell. Just try to connect. This paragraph will address. Suppose that this is a host cell. I am telling it is a which cell? Host cell. And this is the desired gene. Assume it is insulin producing gene. It is desired gene, foreign gene, alien gene. Many names are there. Got the idea? So it is a desired gene. Are you getting my point? So we don't incorporate desired gene directly into the host cell. We don't prefer to do it usually. Reason? Why we are not directly incorporate the desired gene in the host cell? Because in this desired gene who is not there? Oocyte is not there. Origin of replication is not there. And you better know, without oocyte, a DNA cannot replicate. So if this desired gene don't have oocyte and we are adding into which one? Host cell, what happen? These bacteria increase their number in a culture medium, but who can't make their copy? Desired gene not make their copy. So who is going to fail? Gene cloning will not occur. Na? Our desired copy of gene not reach in every bacteria. So that is the reason it is not a correct practice. What we are doing? First we are incorporating, we are introducing the desired gene into a vector DNA. Suppose that this is a vector DNA. Understanding the idea? These vector DNA have what? Basically origin of replication. They have oocyte. So what we are doing here? We are cutting the vector DNA. We are joining the desired copy of gene. We are joining the desired copy of the gene here, and then we are making it with which one? Recombinant vector. In short, I am writing RV, and then we are transferring into the host cell. Got the idea? So what is the purpose of a vector? Because desired gene, foreign gene, alien gene don't have oocyte, so it can't make its many copy. Gene cloning become not possible inside the host cell. So to facilitate the gene cloning, what we are doing? We are joining the desired gene with the vector DNA which have oocyte. Got the idea? And then we are transferring into which one? These recombinant vector into the host cell. So here these recombinant vector is also called chimeric DNA. Chimeric DNA. This recombinant vector is also called which one? rDNA have a other name also, that is what? Chimeric DNA. And now this chimeric DNA enter into the host cell. So these host cell now become genetically modified organism. Because these bacteria earlier don't have, suppose that desired gene is insulin producing gene, now they have the insulin producing gene. Are you getting the idea? Understand? So it become a genetically modified organism. So what is the need of a vector DNA? You got the idea, understand the idea? I hope yes. Now what I am going to talk, I am asking, NCERT ask a question. How, it is an important question to address, how biotechnology, biotech is better than, or genetic engineering is better than traditional hybridization. Hybridization during plant breeding. So you know better, in plant breeding, we are making a cross between two desired variety of plant. So what happened in the plant breeding? Both good and bad character of parent are transferring into the progeny. So two parent are there, male and female parent are there. In gamete of the male or female, both good as well as bad character of the parent will go. Good character also go, bad character will also go. We can't stop the inheritance of bad character as well. But in biotechnology, we can do it. Because in biotechnology, we are incorporating a gene. So what gene we will wish, we will incorporate that only. Na? Are you getting my point? What I want to say? Whatever our choice is there, I want this gene. We will incorporate that gene only. But it is not possible in which one? Plant hybridization. So in such a way, who is better? Who is better? Which one? Biotechnology is better than hybridization. How better? Because how the answer you will give? Because in biotechnology, we incorporate only desired gene, whereas in which one? Hybridization, both desired and undesired character of parent transfer to the next generation. Are you getting the idea? Is it clear in the mind? Now we are going to move ahead. So this paragraph is almost important area is addressed. Now we are going to move ahead. Have a look here. Now I am just going to say you about the construction of first rDNA. How the first rDNA constructed. Construction of first rDNA. So these first rDNA constructed in 1972, and for that you must not forget to give credit to Herbert Boyer and Stanley Cohen. Herbert Boyer and Stanley Cohen. So Herbert Boyer, I will not talk more here. So Herbert Boyer learned a technique. What technique he learned? How to cut the DNA into fragment and how to join two fragments of DNA. That technique learned by whom? Herbert Boyer. Are you getting my point or not? Herbert Boyer learned a technique how to cut the DNA into fragment and how to join two fragments of DNA. Don't forget. Now another scientist was there whose name was what? Basically Stanley Cohen. Stanley Cohen learned a technique. What technique he learned? Cohen, what technique he learned? How to isolate a plasmid from a bacterial cell. And then he learned another technique, how to introduce this isolated plasmid into a host cell. So this technique learned by which whom? Stanley Cohen. How to cut the DNA into fragment and how to join two pieces of DNA. Got the idea? That technique learned by whom? Herbert Boyer. This technique learned by whom? Stanley Cohen. Both decided to work together. Got the idea? And when they work together, a miracle occur in the science. Got the idea? What we was not thinking earlier. We create a genetic modification. First time we creating which type of condition which is not in nature, and we work against the nature. And it become possible in 1972. So how it become possible in 1972? I am just telling you. From here also question is going to ask. So listen it carefully. Give your attention. So I am erasing it. I am writing on this page only. So they have taken plasmid from Salmonella typhimurium. Salmonella typhi in short I am writing. In short I am writing. So this is the plasmid they have taken from which one? Salmonella typhimurium. Got the idea? So who know this? Stanley Cohen know it, how to isolate the plasmid. Now along with them, who is present? Antibiotic resistance gene is present. So this is what basically antibiotic resistance gene. Antibiotic resistance gene is there. This is the antibiotic resistance gene. NCERT not talk anything about the source of antibiotic resistance gene. So don't put your brain here, no one will ask. Sir, from where this antibiotic resistance gene came? Because NCERT is silent here. So they have written what? They have taken plasmid from Salmonella typhimurium. I am making it double-stranded, otherwise you will think like, sir, make what? Single strands of plasmid is single stranded. No, it is what? It is plasmid. And these plasmid have what basically? ORI site. ori site. This is the desired gene. Desired gene is antibiotic resistance gene. Now what they did, they cut the plasmid as well as desired gene with which one? Same enzyme. So complementary sticky create and they join it further. So when they join it further, so it become a which one? rDNA. Got the idea? Later on what NCERT is writing? NCERT writing further, NCERT writing in this paragraph further, कि they have taken a which cell? Bacterial cell who is like the which one? Salmonella typhimurium, and who is here? Escherichia coli. Escherichia coli. Then they transfer this one into which one? Escherichia coli. So when they transfer in the Escherichia coli, these recombinant plasmid enter into which one? Escherichia coli. Got the idea? So here these E. coli become GMO or not? It is a genetically modified organism or not? It is a genetically modified organism. Now earlier, before doing this job, E. coli don't have resistivity against which one? Antibiotic. Now E. coli develop which one? Antibiotic resistance. Got the idea? So it will remain in your mind. This entire story will remain in your mind. Once again, it is important, so listen it. They have Herbert Boyer and Stanley Cohen, how they constructed the first recombinant DNA in 1972. They have taken plasmid from Salmonella typhimurium. Then they have taken antibiotic resistance gene. Plasmid source NCERT is writing, antibiotic resistance gene source NCERT not writing. Then they join the two, then transfer into a bacteria similar like that. That is which one? Escherichia coli. Escherichia coli got developed which one? Antibiotic resistance. Earlier they don't have resistivity for the antibiotics. You got the idea? Understand the idea? I hope it is clear. Right now we are moving ahead. So in such a way, they just construct the first antibiotic resistance gene in 1972. Now give your attention. So this is the story. This part of the story is what basically over. Now, very easy and very important questions going to ask every year in the exam. So we are cutting the DNA. We are taking a plasmid. We are cutting the plasmid by which enzyme? We are cutting it with which enzyme? Molecular seizure. Molecular seizure. Who is acting as a molecular seizure? Restriction endonuclease. Cutting and joining. So cutting done by which one? Restriction endonuclease. So it acts as molecular seizure. Every year almost you will find in some paper and even sometime in Neet you will find we are asking what is the job done by restriction endonuclease? It cut the DNA at a specific site and it break which bond? It break which bond? Phosphodiester bond. It break phosphodiester bond. Who break phosphodiester bond? Restriction endonuclease break phosphodiester bond in the strand of DNA and they cut the DNA into fragment. Here there is no specialized enzyme for breaking of H bond. Got the idea? Now another is molecular glue. These two enzymes is very important to keep in mind. Molecular glue. Who act as molecular glue? DNA ligase. DNA ligase. These DNA ligase join two fragment of DNA. Join two fragment of DNA. Who is acting as a molecular glue? DNA ligase. They join which bond? They join, don't forget these two lines. We find every time in test people doing mistakes, some of the students. They join phosphodiester bond. They join which one? Phosphodiester bond. Got the idea? Who is breaking phosphodiester bond? Restriction endonuclease. Who is joining phosphodiester bond? DNA ligase. So who is acting as a seizure? Restriction endonuclease. Who is acting as a glue? DNA ligase. Don't forget this line also. Got the idea? So they are talking about the, right. Now we are moving ahead. Now further NCERT wrote here, such type of question sometimes they ask in code type of question, like they will ask the 3 basic steps in genetic modification of an organism are. What are the 3 basic steps? So first basic step is what? Identification of a DNA with a desired gene. Introduction of identified DNA into host, and maintenance of introduced DNA into the host and transfer of DNA into progeny. These are the 3 basic steps in genetic engineering. Got idea? Or genetic modification. 2 main principles are genetic engineering and bioprocess engineering. What are the 3 basic steps? Identification of DNA with desired gene, introducing the desired DNA. So suppose that I am telling, suppose that this is DNA, this is DNA, this is DNA, this is DNA. Which is the DNA with the desired gene? So suppose that this is the DNA with the desired gene. Second, we are introducing into the host cell. So first step is here. Second step, we are introducing into the host cell. Now third step is what? We are growing the host cell in culture medium. So many host cell is going to produce. So these are the 3 basic steps we use in the genetic modification. These are very easy thing, but people going to ask sometime. Got the idea? Over. Now we are moving ahead. Now we are going to talk about tools we use in which one? rDNA technology. And in tools, we are going to talk first about which one? Restriction enzyme. We are going to talk about which one? Restriction enzyme. Give your attention. So listen about the restriction enzyme. What NCERT wrote in the beginning of the paragraph. NCERT wrote that in 1963, 2 enzymes responsible for restriction of bacteriophage growth in E. coli were isolated. How many enzymes isolated? 2 enzymes isolated. Got the idea? What this 2 enzyme is doing? Out of the 2 enzymes, one enzyme is protecting the bacterial DNA, and another enzyme cutting the viral DNA. You just listen it in a story format. Here you will also understand. Actually, problem is what? Unless you will not understand the problem, you will not understand the solution. So problem is, this is a bacteria and this is a virus. Okay, I'm just making a rough sketch. Suppose that this is a virus, this is a bacteriophage virus, right? So what happening basically? These bacteriophage virus infect the bacteria, and after infecting the bacteria, they are increasing their population in the bacteria. They hijack the metabolism of bacteria and they are increasing their own population in bacteria. Then what is the problem with this? They are using the raw material of bacteria, resources of bacteria, and at the end what happened? In most of the cases, they are killing the bacterial cell. Lysis occur. So such kind of life cycle of virus is called lytic cycle. So any bacteria want to welcome such kind of guest who came in their house, eating, enjoying, watching TV, and in last when they are going, they are killing everyone. No one want such kind of host, such kind of guest. No host want. So what bacteria is doing? To avoid the infection from this virus, bacteria is releasing 2 type of enzyme. Why 2 type of enzyme? One type of enzyme is protecting the genome of the bacteria. This is the bacterial DNA. So one enzyme protecting the first enzyme protecting the bacterial DNA by adding methyl group. It is called methylation. Methylation. So they perform methylation. It is like a flag to know we are the same, from the same family. Got the idea? So they are creating methylation on the bacterial DNA. This is done by which enzyme? First enzyme. NCERT not wrote the name, but I am writing it is which one? Methylase enzyme. So methylase enzyme protecting the bacterial DNA. Two enzyme NCERT wrote na. One enzyme protecting the bacterial DNA. Another enzyme doing what? Who is coming? Here viral DNA is coming. Here viral DNA is coming. Got the idea? It is viral DNA. Now another enzyme doing what basically? As viral DNA is coming, they are chopping it into pieces. So with the help of one enzyme, what happening? You try to understand a story. Suppose that you are in a which one? Mass fighting between two group of people. Right? Stone paddling you are doing. So you don't know na who 100 came with me. You don't know the face of each 100. So what you are doing? Vaya, from my side who are 100 na wearing the red color t-shirt. So it is a what basically? Tagging na. So I will not paddle the stone on who are wearing the less red. So same happening here. So they are protecting their own DNA by which one? Adding the methyl group, creating methylation done by which one? Methylase, and they are cutting the viral DNA and making them infection free by the second enzyme, and the second enzyme who are cutting it called which one? Restriction endonuclease. Got the idea? Is it clear in the mind? So these restriction endonuclease have power to cut DNA into which one? To cut DNA sequence into which one? Pieces. So when viral DNA going to cut into pieces, what happen? They will not able to multiply, their information get disintegrated, they can't make their replica. So you got the story or not? And of course it is performing in which one? Palindromic region, because there it is going to cut only, na? Restriction enzyme cutting only, na? Getting the idea? Is it clear in the mind? So these are the about the restriction. So many question is going to ask. Let me write the question, then I will give the answer of all these questions. So many small questions are from here. So one small question is, what is the source of RE? RE stands for restriction endonuclease. Question number 1. Right? So what is the answer of the question you will give? Answer of the question is what? Answer of the question is what? Bacteria. Bacteria is releasing the restriction endonuclease enzyme. Next question. Why bacteria release RE? Why bacteria release RE? Restriction endonuclease enzyme. Why they are releasing restriction endonuclease? So answer is, many times this question asked in the exam, and it is also very important for this year. So in defense mechanism. Defense mechanism. Defense mechanism means what? They want to protect themselves from infection of the virus. Got the idea? Why bacteria releasing RE? For defense mechanism. Don't write for genetic engineering. We are exploiting this enzyme for which one? Genetic engineering. So what are the use we are doing for the RE? We are using the RE in which one? Genetic engineering. As a which one? Tools. We are using it as a which one? Molecular seizure. But bacteria using it in which one? Defense mechanism. Got the idea? Now question is there. Next question. How many different restriction endonuclease isolated till now? According to NCERT data, it is what? More than 900 different restriction endonuclease we isolated from above 230 strains of bacteria. Got the idea? Is it clear in the mind? Is it understandable, clear or not? So what is the source of RE? Came bacteria. Why bacteria releasing RE also came? Why bacteria releasing RE? For defense mechanism. What are the use done by, we are using, what are the use human is doing for which one? RE. Where we are using the RE? We are using RE in which one? Genetic engineering. We use RE in which one? We use RE in genetic engineering. genetic engineering. But bacteria use in defense mechanism. So this is the area also over. Now we are going to talk about many RE is there. Now many RE is there. So we are going to talk about nomenclature of RE. Nomenclature of RE. Nomenclature of RE. Got the idea? So I am just telling about the example. Suppose that it is eco R1. This is a restriction enzyme. What is this? Restriction endonuclease enzyme. Restriction endonuclease enzyme. Once we ask a question, many of students made it wrong. So RE is chemical nature. Tell me the chemical nature of this RE. Restriction endonuclease eco R1. So this is chemically what basically? Polypeptide, or you can write which one? Protein. It is enzyme, na? So you have studied already, all enzyme are protein except ribozyme. You have studied about it, na? All enzyme are protein except ribozyme. Is it clear in the mind? So don't put your more brain ki it is either ribozyme or not. Why ribozyme it is not there? Peptidyl transferase is a ribozyme. Ribozyme example is not like many are there. Able to understand or not? So you can think like other than ribozyme, whatever the enzyme is there, it is made up of polypeptide or protein. So chemically polypeptide or protein. But the idea, so now about the nomenclature, what nomenclature is saying? First letter of the name of enzyme represent the genus of the bacteria and it is written in capital letter. Second and third name represent which one? A species of bacteria, and both are written in which one? Small letter. Now R is here basically a strain of bacteria from where it is isolated, and 1, 2, 3 is which one? Order of isolation of enzyme from a specific strain. Order of isolation of enzyme from a specific strain. From a specific strain. Are you getting the idea? So what they are saying, with this name, with this enzyme name, we decoded everything about bacteria. This is the enzyme isolated from. E stand for Escherichia. Escherichia is the genus. Coli, CO, coli is the species. A strain name, RY13 is the name of a strain. And it is the first enzyme isolated from Escherichia coli RY13 strain. It is the first strain isolated from which one? Escherichia coli RY13 strain. So you must not forget, first RE is what NCERT wrote? Hind 2. First RE is which one? Hind 2. But it described 5 years later. NCERT also wrote about, na? First RE is which one? Hind 2, but it is described 5 years later. Understanding the idea? Now RE is over, restriction endonuclease is over. Now where RE is cutting and how RE is cutting and joining, I am telling you. Listen it carefully. Listen it carefully. So restriction enzyme cut the DNA. Wait a minute. I just want to say something more. So I am giving you basic classification of restriction endonuclease enzyme. Restriction enzyme, better to write restriction enzyme. It is of two types, exonuclease and it is endonuclease. If we talk about restriction exonuclease, it cut the DNA at terminal end. cut the DNA at terminal end. And restriction endonuclease, second type of restriction enzyme, cut the DNA at a specific site. A specific site, and this specific site is called a recognition site. This specific site is what? A recognition site. What is the character of the recognition site? These recognition sites are what? Palindromic. Palindromic means what? Same reading frame in both forward and backward direction. In English letter it is Malayalam, or in DNA palindromic sequence I am writing GAATTC, GAATTC. This is a DNA. It is a 6 base pair long palindromic sequence. It is 5' to 3', it is 3' to 5'. Understanding the idea? This DNA fragment is going to recognize by which restriction enzyme? EcoR1. EcoR1 recognizing this palindromic sequence. And where they are cutting, how they are cutting? They are cutting away from the center, slightly away from the center. They are cutting between G and A. So when they are cutting, so it is a palindromic sequence, na? How it is palindromic sequence? We are always reading in which prime? 5' to 3', GAATTC, GAATTC. Are you getting the idea? And they are cutting away from the center, and when they are cutting away from the center, they are creating a single stranded overhang. It is one fragment of DNA become like this, another fragment of DNA become like this. So it is single stranded overhang, that much part. That much part is single stranded overhang. Single stranded overhang, and it is called which one? Sticky end. It helps to join the two different fragments of the DNA. It is a sticky end. So EcoR1 is cutting like this. Every enzyme recognizes a different palindromic sequence. Got the idea? An example I am telling: GGATCC, GGATCC. This is also a palindromic sequence. Is it there or not? 5' to 3', 3' to 5'. But it is not recognized by which one? EcoR1. EcoR1 will not cut here. Who will cut here? Here BAM-H1 will cut. It is also cutting slightly away from the center. Center is which one? This is the center, but they are not cutting in the center. Slightly away from the center they are cutting. And when any enzyme cuts slightly away from the center, what happens? They will create a which one? Single stranded overhang. And this one cut by which enzyme? BAM-H1 enzyme. This one cut by which enzyme? EcoR1 enzyme. So what the moral of the story is there? Different restriction enzyme cut at the different recognition site. It's not like any enzyme will cut at any recognition site. So you got the idea? This area is over, cutting, and so now we are coming on. So cutting is done here. Restriction enzyme is done here. Okay. How cutting? Okay. Is it clear? Cutting and joining you want to learn as well. So I am telling cutting and joining. Wait a minute. It will take another 1 minute. We will fix it also. So the things become easier for us later on. Have a look. Now give your attention. Suppose that this is the vector DNA. Suppose that this is the vector DNA. And here it is which one? Desired gene. Desired gene. Now our target is to join the two. Have a look here, and it is important you try to understand. So we are using same restriction enzyme to cut the both desired gene and vector DNA. So we are taking which one? EcoR1. We are taking EcoR1 to cut. So EcoR1 will cut on the same position in both vector DNA and desired gene. So it is going to cut at which segment? GAA, TTC, GAA, TTC. This is which DNA? This is the vector DNA. So here I am taking which one? Desired gene. Here it is which one? GAA, TTC, GAA, TTC. Just try to understand. Is it clear? Right. Now what we are going to talk, we are using the echo R1 to cut the both. So echo R1 will cut here like this. It will cut here like this between G and A. Okay, after cutting, what will the possibility here? G, C, T, T, A, A. It is one fragment. NCERT showing only one fragment. But how many are there? One side is another. It is which one? A, T, T, C, G. Got the idea? If it is a circular DNA, only one fragment came. If it is a linear vector, how many fragments came? 2 fragments came. Are you getting the idea? Now, here just see the things. Here what will come? Another color. G, C, T, T, A. Here who will come? A, T, T, C, G. It cut like this. Who cut it? Restriction enzyme. Which bond they have broken? Phosphodiester bond. Now we are using which enzyme? Ligase. Now we are using which enzyme? Ligase. So now when we use the ligase, DNA ligase, 2 possibilities are there. One possibility is self-ligation. Vector DNA sticky end once again joined with vector DNA. Both are sticky end, na? Both are complementary, they join. So it will form non-recombinant. Here, desired DNA fragment, desired DNA join, it is which one? Non-recombinant. Possibility are getting the things or not? They rejoin together. So when this will rejoin with this, any new copy join together? No. When this rejoin with this, it is possible that TTA pair with once again ATT. What bond required to do? Phosphodiester bond formed by which one ligase? Here also phosphodiester bond formed by ligase. This is one possibility. Is there or not? So when we are using the ligase, it is very important to understand, when we are using the ligase, possibility number 1, self-ligation, means non-recombinant form. Another non-recombinant form. Another possibility is what? Another possibility is there, vector DNA join with desired DNA. In that case, who will form? Recombinant will produce. In that case, who will produce? Recombinant will produce. So here, see, here, G, C, T, T, A, and this is which one? A, T, T, C, G, and here ligase is joining the 2, then it becomes which one? Recombinant DNA. So how many learning came here? 2 learning came here. When we are cutting with the restriction endonuclease and joining with the ligase, there are 2 possible outcomes is there. If self-ligation occur, who will form? Non-recombinant. If cross-ligation occur, vector DNA ligate with desired DNA, who will produce? Who will produce? Recombinant will produce. You got the idea? So this entire story is over. How to cut, how to join, this entire story is over. Give your attention. So this entire story is over. How it is cutting, which bond is there, how it is joining. Got the idea? Is it clear? Now I am going to tell you a basic idea about which one? Gel electrophoresis. Gel electrophoresis. Give your attention. Just have a look about gel electrophoresis. So gel electrophoresis is a technique to separate the charge particle. These charge particle here is what? DNA. We are talking in the term of DNA, but we can also separate RNA through it. We can also separate protein through it. Here we are telling about the separation of DNA. How we are using the gel electrophoresis? These are the sample loading well. And in this sample loading well, what we are doing, we are adding here the DNA. Suppose that we are removing the DNA from the human cell and we added the DNA into the sample loading well. Later on, this sample loading well is present toward a negative charge. That is which one? Cathode. At cathode end it is there. And here it is which one? Negative, positive charge. It is anode end. Got the idea? Now here we are using restriction enzyme. Any restriction enzyme, suppose that we are using BIMH1 or EcoR1, which enzyme you want to use to cut. Got the idea? So we are using it. So what restriction enzyme will do? What restriction enzyme will do? Let me tell you. This is the DNA. This is the DNA. Now you just try to understand. Here it is the recognition site for EcoR1. This is one recognition site, it is another recognition site, it is another recognition site. These are the recognition site for which one? EcoR1. So when we add the EcoR1, these EcoR1 will cut at the wherever who is there, recognition site with there. So one fragment here, one cut here, one cut here. So DNA arranged according to the fragment. Because all the eco are not present on the equidistance, fragment length of the DNA after restriction digestion will be which one? Different. So it is the longest fragment, it is second longest fragment, and it is which one? Third longest fragment, and it is the shortest fragment. Are you getting the idea? So what we are finding, fragment length which is producing after restriction digestion are not same. Why not same? Because everyone not present on the equidistance. So now what happening? Every which one? Recognition site not present on equidistance. So now when we are adding restriction enzyme, it is chopping the DNA into fragment, and the fragment piece is different. Now in this plate, who is filled? Agarose gel is filled. Who is filled? Agarose gel is filled. Agarose agar obtained from which one? Seaweed, Geladium and Gracilaria. Are you getting the idea or not? Okay. Okay. Agarose. Right. So here it is creating which one? Pores. You have seen the bread. In bread pores are there now. So here pores are even bigger than that. Are you getting the idea? So they have a sieving effect. So what happened when DNA cut into fragment, restriction digestion occur, and when we flow the current, DNA particle move like this. What you will find, these shortest fragment moving which one? Farthest. And the longest fragment moves slowest. Question arises, why the fragment is moving? Reason is, it is DNA is negatively charged, and this negatively charged DNA present at which one? Cathode. Sorry, present at which one? Cathode. Negative charge. Negative repulsion, negative positive attraction. So movement of the fragment occur due to charge. Negative repulsion, negative positive attraction. Movement is due to charge. But different fragment length moving with the different rate. Differential movement. Differential movement you are getting. Different fragment moving with the different rate. It is due to size or molecular weight. As a bigger size of the fragment, it faces more resistance to pass through sieve. Sieve is there, pipe is there, tunnel is there. Suppose that you are crawling through a tunnel and a 5-year baby also crawling through a tunnel. Who can crawl faster and go another side? 5-year baby, because they are facing little resistance. Are you getting the idea? So differential movement of fragment is due to which one? Size. Movement is due to which one? Charge. If it is a neutral molecule, it can't move. Are you getting the idea? Negative repulsion not occur. So they can't migrate toward which one? Anode. Getting the things or not? So here it is a charged particle is moving. Getting the thing? DNA is a negatively charged particle. That's why it is moving. in the gel plate. Differential movement is due to which one? Difference in the size. Are you getting the idea? Clarity came in the mind, right? Now what NCERT wrote? NCERT wrote further here that these what DNA bands are not visible, so we are staining it with which one? Ethidium bromide. Ethidium bromide. It is a question. Every line is a question here. We are staining with which one? Ethidium bromide. Then visualize with which one? Ultraviolet rays, UV rays, and when we visualize, it is giving which color? Bright orange color. Bright orange color. Got the idea? Now what NCERT wrote? Later, NCERT closed the story pathetically, and I am doing the same here because no one will ask in the exam. That's the reason what NCERT did, I am doing. What NCERT is telling? Then we isolate desired DNA fragment by elution. NCERT not talked about how to know who is desired DNA fragment. Here thousands of fragments are there, lakhs of fragments are there. Out of that, anyone is desired DNA fragment. NCERT not addressed it. How to know who is the desired DNA fragment? Simply NCERT telling what? We isolate the desired DNA fragment with the which one? Elution. So here it is the gel plate. We are cutting the gel block. Suppose that this is desired DNA fragment. We are removing this part of the gel block like a piece of cake, and then washing the gel and then isolating the desired DNA. So this is about the gel electrophoresis NCERT wrote. I hope you understand what is gel electrophoresis. Is it done? Gel electrophoresis is over. Now we are moving ahead. Now we are going to talk about the cloning vector. Now we are going to talk about which one? Cloning vector. Give your attention about the cloning vector. So here we are going to take the example of this one. PVR 322 is a which one? Cloning vector. Are you getting the idea? Just if it is possible to somehow more zoom in, I am trying to do it. So this is about a cloning vector. Have a look now. Just give me a minute. So what is the character a cloning vector must have? It is very important to know. Cloning vector must have these three characters. Character number one, it must have ORI site, origin of replication. Because desired gene don't have ORI site, so we are attaching it with which one? Cloning vector. So it must have ORI site. And be preferable to take such cloning vector who have high copy number. So rate of replication of the plasmid here decided by which one? ORI site. Such plasmid is also there in X minute, it is making 200 copies. Such plasmid is also there in X minute, they are making 20 copies. Such plasmid is also there, we are making 10 copies. So we prefer which one? Who are making more copy. Because more copy number, more rate of replication means what? More faster gene cloning. So we prefer the ORI site with which one? High copy number. ORI site is origin of replication. It is essential for DNA replication to occur. Second important quality a cloning vector have is what? A recognition site. A recognition site. Second property a cloning vector must have is what? A recognition site. Recognition site is what? It is palindromic sequence. Palindromic sequence. Because why recognition site is important? Because restriction endonuclease not cut any other place other than recognition site. So if the recognition site is not there, you cannot cut the DNA into fragment. And be careful, there must be only one recognition site for one enzyme. If it is more than one recognition site for one enzyme, what happens? DNA will cut into fragment. So there must be one recognition site for one enzyme. And third quality must be present in a cloning vector is it have selectable marker. Selectable marker gene. Selectable marker gene. What this selectable marker gene is doing? It help to differentiate between first transformant and non-transformant, and second recombinant and non-recombinant. This is done by which one? Selectable marker gene, to differentiate between transformant and non-transformant and recombinant and non-recombinant. Now we are going to talk about, so you got the idea, three property must be there in a cloning vector: ori site, recognition site, and selectable marker gene. Now we are moving ahead. Give your attention. So here we are talking about an example of a cloning vector. The name is what? PBR322. In PBR322, P stand for plasmid, B stand for Bolivar, R stand for Roderick, name of scientist who constructed it. So it is not a natural plasmid present in a bacteria. It is synthesized in a laboratory from a normal plasmid, modified in the laboratory. A normal bacterial plasmid modified in the laboratory by whom? Bolivar and Roderick. And 322 is the batch number of the plasmid. Batch number means in which lab it formed, that is the batch number of that plasmid. Now, how many things present in this plasmid? In this plasmid, this is the ori site. Please don't forget the labeling. This diagram is very important. This diagram is very important. Take care about the every labeling. This is the ori site. This is ROP region, this segment of DNA. This is responsible for application of plasmid. In this region, there is a recognition site for PVU2 enzyme. Then from here to here, it is the tetracycline resistance gene. And in tetracycline resistance gene, there is selectable marker. There are tetracycline gene used as a selectable marker. It is used as a selectable marker. And in between this tetracycline resistance gene, there are two recognition sites is there. One for enzyme BAMH1, another for enzyme SAL1. Don't forget, in tetracycline resistance gene, resistivity site for BAMH1 and SAL1 is there. This is another selectable marker gene, ampicillin resistance gene. Got the idea? So in ampicillin resistance gene, there is the recognition site for two enzymes. One is PVU1, another is PST1. Got the idea? Now, here in this region, three enzyme recognition site is there. One for EcoR1, another for CLA1, another for HIND3. So in this PBR322, one ori site is there, two selectable marker gene is there, and 1, 2, 3, 4, 5, 6, 7, 8 recognition site for different enzyme is there. But be careful, there is not a single recognition site for the which one? Same enzyme. Two enzymes, same enzyme. Everyone for which one? Different enzyme. If we use BAMH1, it will cut here only. If I use PST1, it will cut here only. Are you getting the idea? Understanding the idea? Is it? If we suppose that here also BAMH1, here also BAMH1, it will cut into two pieces. Na? Got the idea? Understanding the idea? Clarity came? Right? So this must be a character present in which one? Cloning vector. Now we are going to talk about screening. That is very important part where students struggle. Let me talk about it. Let me talk about it. Have a look now. So now most important part of the chapter is there. So who are not attentive, please give your attention now. This is going to help you and they are going to somehow release your stress. Because I know as a teacher, student struggle in which area? This area. So now look here. I am taking PBR322. PBR322. It is a vector DNA. Now I am taking a desired gene. Desired gene. This is PBR322. Give your attention, everyone. PBR322. Now I am using enzyme which one? Restriction endonuclease. Then I am using enzyme which one? Ligase. DNA ligase. So how many possibility are there? Two possibility. Why I am telling? Here, two possibility. Self-ligation, cross-ligation. Got the idea? So two possibilities there. So I am using which enzyme, which restriction enzyme? BAM H1 I am using here. I am using BAM H1 here. Are you getting the idea? So for your understanding, it is ORI site. Here it is tetracycline resistance gene. This area is which one? Ampicillin resistance gene. And suppose that this is recognition site for which one? BAM H1. It is recognition site for BAM H1. It is which one? BAM H1 recognition site. So when we are using restriction enzyme BAM H1 and DNA ligase, how many possibilities are there? Two possibilities there. One is self-ligation. I already explained, when ligase going for join the vector DNA together or desired gene together, self-ligation. And when vector DNA join with desired gene, it is cross-ligation. Are you connected with me or not? Two possibilities there, self-ligation and cross-ligation. Now if self-ligation occur, it will form which one? Non-recombinant. In short, I wrote NR. If cross-ligation occur, who will form? Recombinant. I am writing R. Now, I am writing here in NR, I am asking a question from you. In non-recombinant, ampicillin resistivity or tetracycline resistivity there or not there? It there. Why it is there? Because no new gene added. In non-recombinant, no new gene added. Na? In vector DNA, desired gene not added. That's the reason we are telling non-recombinant. So when new gene add, then insertional inactivation occur. When a new gene added between old gene, what happened? Insertional inactivation of old gene become occur. Old gene become inactive and new gene become active. Are you getting the idea or not? But here, no new gene is added in non-recombinant. So why ampicillin resistivity and tetracycline resistivity will go? It will not inactivate, na? Because nothing is added here. Now when we are using which one? In a recombinant, what we are finding? In recombinant, ampicillin resistivity is there. You can say, sir, why NCRT? Recombinant means which one? New gene added. And tetracycline resistivity is not there. Why tetracycline resistivity is not there? Because we are using which enzyme? BAM H1 enzyme to cut. So BAM H1 enzyme cut in between which gene? Tetracycline resistance gene. Because you just see the PBR322, their recognition site present between which one? Tetracycline. And when ligase will join, the new gene will add between which gene? Tetracycline resistance gene. So whenever new gene will add, when we using which enzyme? BAM H1 enzyme, whenever the new gene will attach between which one? In between tetracycline resistance gene. Are you getting the idea or not? So who become inactive? Tetracycline resistance gene become inactive. Insertional inactivation of tetracycline resistance gene will occur. Did you connect it, understood or not? Now I am asking a question in different way. If I am using which enzyme? PST enzyme. So PST enzyme cut between which one? Ampicillin resistance gene. So when PST enzyme we will use, new gene add in between which one? Ampicillin resistant. So recombinant lose the resistivity for which one? Ampicillin. Conditional, we are using which enzyme? PST1. If we are using BAM H1, new gene add between which one? Tetracycline resistance gene. Are you getting the idea? Anyhow, so what we find? NCRT told every story in the reference of which enzyme? BAM H1 enzyme. So what we are telling? Non-recombinant have resistivity for both antibiotic and which one? Recombinant have resistivity for ampicillin, not for tetracycline. Now just look here, give your attention. Now I am taking a culture medium. In this culture medium, there are millions of bacteria is there. and I am adding here, which one? I am adding here plasmids, but we are doubtful कि either this plasmid is recombinant or not recombinant. We don't know na. When new gene added, they will not say na I am here. Color will not change na here of the gene. Getting the idea? So it is very. So here we are using which one? Plasmid, but we are doubtful it is recombinant or non-recombinant. Another challenge is what? Who is not ready to accept this plasmid? Host cell. Host cell. This is host cell. Host cell is saying no, 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 no. I am not interested to take this plasmid. Why, bhaiya? Reason is plasmid made up of DNA. DNA is which one? Negatively charged particle. And in cell, plasma membrane is there. Plasma membrane is there. That have which one? Lipid bilayer. And this lipid bilayer, phospholipid is showing hydrophobic nature. So these hydrophobic nature of the phospholipid part, getting the things or not allow the hydrophilic DNA to enter. Plasma membrane usually not love to entertain whom? Charged particle. Are you getting the idea? Especially the lipid bilayer part. So they are telling I am not interested. This is another problem. So how these plasmid will enter in the host cell? NCERT wrote na some method. NCERT wrote. First method they wrote what? We are making the host cell competent. How to make the host cell competent? Connect with the line of NCERT I am telling. So that NCERT is writing, we treat the which one? Culture medium, host cell, and what is there? Plasmid is there in the culture medium with which one? Divalent cation. We added so many divalent cation there. Calcium and we are adding. So when we treat it with the divalent cation, what NCERT wrote? It increased the efficacy of entry of plasmid into the host cell. Another thing what we are giving, shock treatment. In shock treatment, what is there? First it is placed in, bacteria is placed on ice. Then we are giving at heat shock at 42 degrees Celsius. Then further we are shifting into ice. So it is called shock treatment. We are shifting from ice to heat at 42 degrees, then into ice. And after that, what happened? Who accepted the plasmid? Host cell accepted the plasmid. Now here million of host cell is there. In which host cell it enter, we don't know na. So in culture medium, what tension we have? So here it is culture medium is there. In culture medium, two category of bacteria is there. Two category of bacteria is there. Listen it very carefully. It is the heart of the chapter. Culture medium, two category of bacteria is there. One is non-transformant. One is non-transformant. Another is transformant. Transformant. Are you getting the idea? So if we talk about non-transformant, what is the meaning of non-transformant? I am writing the meaning. That bacteria where plasmid not enter. Where plasmid not entered. Is called non-transformant. Transformant are what? That bacteria where plasmid enter. That is called transformant. Are you getting the idea? So transformant is of two possible type. If plasmid enter, we are not sure na plasmid is either recombinant or non-recombinant. There are possibility of recombinant also, possibility of non-recombinant also. So two possible type of transformant is there. One possibility is non-recombinant, another possibility is recombinant. Did you understand here? Now I am writing here. In case of non-transformant, I am writing for everyone. Ampicillin resistivity, tetracycline resistivity. No, no. Why no, no? Because NCERT wrote in normal E. coli. Who is normal bacteria? Non-transformant bacteria. They are normal E. coli and they have no resistivity for any antibiotic. It is just pop up the diagram written in NCERT. Any antibiotics. They have no resistivity against any antibiotic. Now if we talk about non-recombinant, ampicillin resistivity, yes. Tetracycline resistivity, yes. Now recombinant ampicillin resistivity, yes. Tetracycline resistivity, no. Conditioning, we are using which enzyme? BAM-H1. Done. Now in culture medium, I am adding whom? In culture medium, I am adding ampicillin. In culture medium, what I am adding? Ampicillin I am adding. You help me, who will die? You help me, who will die? When we add the ampicillin in the culture medium, who will die? In culture medium, I am adding ampicillin. Ampicillin resistivity not present in non-transformant. It is there in both non-recombinant and recombinant. Got the idea? So who will die? Non-transformant will die. So we killed all that bacteria where plasmid not enter. So here can we say ampicillin resistance gene act as selectable marker to differentiate between transformant and non-transformant. Can I say this line? Who is transformant? Where plasmid enter? In which bacterial cell plasmid enter? In which bacterial cell plasmid not enter? It is done with the help of which one? Ampicillin resistance. Ampicillin resistance. Got the idea? Now we are separating the living bacteria from the dead bacteria. Suppose that this one plasmid enter in this one bacteria. We added only one plasmid, na? So only one become transformant, na? Remaining all they are what? Non-transformant. All bacteria died. How many are surviving? Only one. So we separated these living bacteria. Now how to check? It is recombinant or non-recombinant. It have desired gene or not desired in the plasmid. Plasmid came, it is okay. But recombinant plasmid came or not, we don't know, na? So for that we use our brain. We not directly apply which one? Tetracycline. First we are allowing these bacteria to grow and make 2 copy of it. In one paper we ask na how many replica plate we are using to check the recombinant. 2 replica plate. Why 2 replica plate? Now one bacteria converted into 2 bacteria. Both are same bacteria na. So I am taking 2 replica plate. In one replica plate I am adding one bacteria, another bacteria in this replica plate. Both are transformant, it is for sure. But we don't know it is recombinant or non-recombinant. How we make it 2? Be growing in the culture medium, after some time binary fission occur, 2 bacteria form. Now 2 bacteria are same, exactly clone, both are same. Now what we are doing, we are adding tetracycline. It is called replica plating experiment. Now we are adding tetracycline on this. And suppose that after tetracycline using, and it is died now, means it was what? Recombinant. You kill the recombinant. Thanks God we have another copy. If we don't make it replica plating, 2 copy, we kill that recombinant, na? But we killed one recombinant. If one killed, it is clear it is what? Recombinant. I have a 1 copy more, na? I am not adding tetracycline here. Now we become very confirmed, na? Ki if it is recombinant, it is also recombinant, because it is dividing and making it 2, and we are using this. Got the idea? So here tetracycline resistance gene help to differentiate between which one? Recombinant and non-recombinant. Ampicillin resistance gene help to differentiate between which one? Transformant and non-transformant. Is it clear or not clear? Is it done? Can we move ahead? I am just moving ahead now. Now NCERT further wrote. NCERT further wrote that it is a cumbersome process. It is a difficult process. Nowadays, a very easy method is also there to differentiate between recombinant and non-recombinant via color change only. Via color change only. How? Let me tell about that. So here I am taking a plasmid. And in this plasmid, this is a In this plasmid, this is a selectable marker gene. And the name of gene is LAC-Z gene. LAC-Z gene. Now, what I am doing, here we find it is a recognition site for a specific enzyme. It is a recognition site between LAC-Z gene. Now what we are adding here? We are adding the restriction enzyme. We are adding which one? Ligase. We are adding which one? Desired gene. How many possibilities there you better know? One is self-ligation. Means vector DNA joined with vector DNA by ligase. Another is cross-ligation. If self-ligation occur, non-recombinant will produce. If cross-ligation occur, recombinant will produce. Means non-recombinant means no new gene added between LAC-Z gene. So who remain active? LAC-Z gene remain active because no new gene added. Here what happened? LAC-Z gene become inactive. Why it become inactive? Due to insertional inactivation. Because when a new gene added between an old gene, suppose that here new gene added, they attach between which one? LAC-Z gene only. Now suppose that here LAC-Z, in between LAC-Z, here new gene added. So who become inactive? Old gene, LAC-Z gene become inactive and this new gene become active. Got the idea? So here LAC-Z gene become inactive. Here LAC-Z gene active in non-recombinant. Who will produce? Here beta-galactosidase will produce. You know na LAC-Z producing which one? Beta-galactosidase will produce. Here no beta-galactosidase. Beta-galactosidase. Are you getting the idea? If beta-galactosidase produce, how to check beta-galactosidase produce? So when we add chromogenic substances, what is the name of it? X-gal. No one will ask X-gal. Chromogenic substances. Beta-galactosidase react and giving which color? Blue color. So chromogenic substances reacting with beta-galactosidase and giving blue color. When we added here which one? Chromogenic substances. Who is not there? Beta-galactosidase is not there. Reaction not occur. So it is colorless. No color change occur. So when blue color colony is coming, we can say na it is non-recombinant. When colorless colony is coming, we can say na it is recombinant. So simply by identify the change in the color, we can find about what? Either it is recombinant or non-recombinant. But this is not helping to differentiate between transformant and non-transformant. This is only helping to differentiate between which one? Recombinant and non-recombinant. So these screening part is over. What are the most important area of this chapter is over. Now in another 5 minutes we are going to finish this chapter. Have a look now. Have a look now. Now we are going to talk about this paragraph. What is important in this paragraph? Let me say. So, vector for cloning gene in plant and animal. Got the idea? So, NCERT wrote about two things. Don't forget about agrobacterium. I'm not writing, I'm saying. Agrobacterium tumefaciens is a soil bacterium. And it is a natural genetic engineer. Got the idea? How it is a natural genetic engineer? It infected dicot plant. After infecting a dicot plant, it is causing a disease in the dicot plant that is called which one? Crown gall tumor. Got the idea? Crown gall tumor. What happened in this disease? It converts a normal plant cell into a tumorous cell. And tumorous cell release a specific chemical which is used by bacteria. So see na, bacteria enter in plant, modified the plant, instruct the plant to make a which one? Chemical, and that chemical used by whom? Bacteria. So this is like it is acting as a natural genetic engineer. So examiner will ask कि TI plasmid is present in. TI stand for tumor inducing plasmid. So this is a TI plasmid. TI plasmid. This is ORI site, and here a DNA they are called tDNA. So this is tumor inducing plasmid. This tDNA convert normal cell into tumorous cell. And these tumorous cell release a special chemical which is used by bacteria. Now what we are doing nowadays we are using this TI plasmid as a vector during genetic engineering. You can say, how can you use, sir? Because it have a tDNA. So it will convert normal cell into tumorous cell. So we are not using normal TI plasmid. We are using which one? Disarmed TI plasmid. Disarmed means which one? We are removing which part? Disease causing part. tDNA. Now if tDNA is not there, it will not cause tumor. Na? In place of tDNA, I will use this our choice of gene to transfer into which one? Host cell. So nowadays, TI plasmid use extensively. Keep this line in mind. To transfer desired gene in which cell? Plant cell. So it is used as a vector to gene transfer in which cell? Plant cell. Which one? Disarmed TI plasmid. Understand? And TI plasmid present in, this is also a hot question, TI plasmid present in Agrobacterium tumefaciens. Agrobacterium tumefaciens. Got the idea? Now NCERT wrote about another thing, that is which one? Retrovirus. Retrovirus. What NCERT wrote about the retrovirus? Let me tell you. Disarmed retrovirus used as disarmed retrovirus used as vector for gene transfer in animal cell. Animal cell. Got the idea? Otherwise, some retrovirus also is which one? Carcinogenic. It converts normal cell into which one? Cancerous cell. So we are removing that cancer causing part, who convert normal cell into cancerous cell. That part of gene we are removing, and then we are using the retrovirus for gene transfer and treatment of which one? Many diseases like which one? SCID. In treatment of severe combined immunodeficiency, we are using which one? A retrovirus as a vector. Are you getting the idea? Understanding the idea? So for animal cell gene transfer, don't forget retrovirus. For plant cell gene transfer, don't forget TI plasmid, but both are disarmed. Another method of gene transfer, NCERT wrote microinjection. Microinjection is a method of gene transfer in which one? Animal cell. We are injecting the DNA through micropipette. It is microinjection. Microinjection in animal cell. And microprojection, biolistic in plant cell. In biolistic, what we are doing? We are taking DNA coated with tungsten and gold and bombarded on the plant cell. We are using microinjection in which one? Animal cell. Microprojection in which one? Plant cell. That is called biolistic. So this area is over. Now another area we are moving. Give your attention. Competent host cell and everything over, that we dealt. Now here it is. Here it is what basically? Process of recombinant DNA technology. Right. Last year from here question came in Neet exam. And what are the important things you just keep in the mind in the process of rDNA technology is, first process is what? Isolation of desired DNA fragment. In rDNA technology, first what we are doing? Isolation of desired DNA fragment. Then Wait a minute. Sorry. No. So first we are going for isolation of DNA. Then we are going for fragmentation of DNA by restriction endonuclease. First, second, third, we are going for isolation of desired DNA fragment. Third, fourth, then ligation of desired DNA fragment into vector DNA. Fifth, transferring of the recombinant DNA into host. And sixth, culturing of the host cell into a medium at large scale and extracting the foreign desired product. So this is the what basically steps of our DNA technology. I am telling you entire steps in 1 minute. In 1 minute, have a look. So if we talk about, have a look here, if entire picture will come. Suppose that I want to produce insulin. So first job is what? We will want insulin producing gene from human insulin I want. So I am taking a human cell. From human cell nucleus, DNA is there. I have isolated the DNA. It is the DNA. First job over. Second job, what we did? This is the DNA. I am cutting the DNA. I am cutting the DNA by which enzyme? Restriction enzyme. It is second step. We are using RE to cut the DNA into fragment. Third step, what I will do? I will isolate the desired DNA fragment. This is the third step. Fourth step, what I will do? I will take a vector DNA and ligate the which one? And ligate the which one? Desired gene into which one? Vector DNA. Who produce here? rDNA produce. It is the fourth step. Fifth step is what? We are taking a host cell and we are transferring into the host cell. This is the fifth step. Are you getting the idea? This is the fifth step. This is the fifth step. Fifth step. Got the idea? Later on, in last step, what we are doing? We are taking a which one? Culture. What happening? Okay, you know it. We are taking a which one? Host cell and transferring. We already did the fifth step. We are culturing it now. We are making multiple copy of it. When we are multiple copy making, what happens? Gene cloning occur. Everyone will produce the desired product. Got the idea? Now isolation of DNA. How to do the isolation of DNA? What NCERT wrote? Just a minute. So isolation of DNA. What textbook is writing? So in isolation of DNA, we are first bursting the cell, rupturing the cell. That word is not written in NCERT. We are rupturing the cell, by bursting the cell. Cell will break. What the idea? Then we are using so many different enzymes, like protease for digestion of protein, lipase for digestion of lipid, RNA for digestion of RNA. If it is plant cell, cellulose and pectinase. Got the idea? To digest which one? Cellulose and pectin. If it is bacterial cell, we are using lysozyme. Are you getting the idea or not? We are if it is fungal cell, we are using chitinase. We are using every enzyme other than DNA. So who get digested? Everything get digested other than DNA. Now what we are doing, we are adding chilled ethanol. It will precipitate the DNA. And by taking a rod, glass rod, we are spinning it and removing the DNA by process of which one? Spooling. Spooling. So spooling, you don't forget. Isolation of DNA is spooling. Isolation of DNA fragment from gel is called elution. So this is about the isolation of DNA. Got the idea? Later on, we are going to move ahead. Have a look. Cutting off DNA, we already know. I will not talk about. Now we are going to talk about PCR. What is PCR? What is PCR? PCR is a method of gene amplification in vitro. Gene amplification in vitro. In vitro means outside the cell. Right? We are doing in a machine called thermocycler. So what happening in PCR? The very first event in the PCR is denaturation. Very first event in the PCR is what? Denaturation. And we are doing it at high temperature like greater than 92 degrees Celsius or in the range of 92 to 94, 96. Different book wrote different story. So very high temperature, at very high temperature, who will denature? DNA will denature, hydrogen bond will break. So they are the hydrogen bond who break here. Got the idea? Later on we are doing annealing. We are doing it somehow lower temperature. It is 56 degrees Celsius. And in annealing, what we are doing, we are attaching the DNA primer in 5' to 3'. DNA primer in which direction? 5' to 3' direction. Are you getting the idea? So annealing is done by DNA primer. It is complementary to the template stand of the DNA. Later on we are using a Taq polymerase enzyme. Taq polymerase we are isolating from which one? Thermus aquaticus bacteria. These are a bacteria who survive at very high temperature. Got the idea? More than 90 degrees Celsius. Why we are taking especially the enzyme from there? There is a reason. And reason is what? Reason is what? Because entire process of PCR we are doing at high temperature. See na, denaturation we are doing at more than 92 degree. You will ask, sir, but extension we are doing at which one? 72 degrees Celsius. So we adding everything all together, na? So we must use enzyme who even survive at which temperature, not denature at which temperature, 92 degrees Celsius. That's the reason we are taking the enzyme from Thermus aquaticus, and enzyme is Taq polymerase. It is a type of DNA polymerase. It perform extension, and after extension it will what? It will going to make complementary strand of the DNA. One DNA converted into two DNA. If we make it multiple copy, what happen? If we go for repeat it at how many cycle? How many cycle? If we repeat for how many cycle? Tell me. 30 cycle. It is getting around 1 million copy of the desired gene. PCR nowadays use in which one? Diagnosis of the diseases also. Getting the idea or not? That is in the zoology side. So PCR is also over. Now after that, these already you know. Now obtaining foreign gene product. Here I am telling you. So we are going to talk about obtaining foreign gene product comes under downstreaming. Downstream processing. Downstream processing. So in downstream processing, what we are doing? We are doing for isolation of product. After isolation of product, we are going for purification of product. Then we are adding preservative to increase the shelf life. Expiry date based on that, na? Preservative. Now we are going for which one? Testing. Trial. Depend upon product to product. What the idea? Then we go for license from the government. So these all comes under which one? Downstream processing. Before marketing, everything what we do with the product is called what? Basically downstream processing. And lastly, I am telling 1 minute about which one? Bioreactor. So what is bioreactor? Bioreactor is a metal vessels where we are producing the desired product in large amount. Got the idea? In a sterilized environment, in a contamination-free environment, and we are performing it at a specific pH, a specific temperature, and a specific aeration. So everything under a control conditions present in which one? Bioreactor. Two type of bioreactor NCERT wrote here. One is simple stirred tank bioreactor, this one. Another is a sparged tank bioreactor. See, you just go for the labeling of this diagram. Nothing is going to ask more, and I am not going to talk out of NCERT, otherwise many type of bioreactor are there. Got the idea? It is not required. So this chapter is over. Now we are going to move on the next chapter. Next chapter. So let's start about the next chapter. After finishing the biotechnology, we will take a small break. Both chapter of the biotechnology. Second chapter is easier, so we will finish in 20 minute, 25 minute maximum the second chapter. Okay, so we are going to talk now about the application of the biotechnology. Second chapter of the biotechnology. That is what? Application of biotech. It is a smaller chapter. So we will invest more time in bigger chapter and less time on a smaller chapter. So application of biotechnology. No need. Nothing. So just give me a moment, 1 minute water break, then we are starting with application of biotechnology. Hanji. Application of biotechnology in human welfare. This is the agenda of the chapter. So first you must know about what are the three critical. So where the biotechnology is used nowadays. So biotechnology nowadays used in almost every area in our life. It used in medicine. You can say how it is in medicine. It is used in medicine in diagnosis of diseases. PCR. Got the idea? Early diagnosis of diseases. Biotechnology have a role now. When COVID came, who was helping? PCR was helping. RT-PCR was helping. Either we are COVID positive or COVID negative. Getting the things or not. So it is have a role in diagnosis. It have a role in therapy, treatment, SCID treatment, SCID treatment, by gene therapy. It is done by which one? Biotechnology. And even nowadays, so many recombinant medicine are there in the market. Like recombinant insulin are there. When you go for vaccination of children or your vaccination when you was smaller, and even nowadays, suppose that children of your age are going for vaccines for which one? Cervical cancer. That vaccine developed through which one? Biotechnology. These are recombinant vaccines. Got the idea? So it have a role in formation of the vaccines, formation of recombinant insulin, right? Treatment of diseases like SCID. So medicine, it have a great role. Another role, biotechnology have a role in agriculture that we will deal in detail. Where biotechnology is used in agriculture. Got the idea? Other area also biotechnology helping us like energy production, waste management, waste treatment, cleaning of environment, bioremediation, bioenergy formation like formation of biodiesel. So that NCERT wrote कि nowadays biotechnology helping us in which one? Have a role in therapeutics, diagnosis, genetic modified crop in agriculture, bioremediation. Bioremediation means what? Cleaning of environment. An example by Pseudomonas putida, waste treatment, and even energy production. Energy production is what? Biodiesel. But what are the three critical research area of the biotechnology? People will ask. So these are the line of NCERT you keep in mind. First critical research area is provide best catalyst in the form of improved organism or a pure enzyme. Creating optimum environment through engineering for enzyme to act. And another is what? Downstream processing. These are the three critical research area of the biotechnology. Now we are going to talk about biotechnology in agriculture. So first, before moving on agriculture, biotechnology role in agriculture, we are just going to discuss about agriculture in our country. We are going to discuss about what? Agriculture in our country. So how many type of agriculture we are practicing? There are three type of agriculture is in practice nowadays. That one is what? Organic farming. Another is agrochemical based agriculture, and third is agriculture by using genetically modified organism. Mostly nowadays we are practicing which agriculture? Agrochemical based agriculture. So what is organic farming? Before Green Revolution in our country, only one agriculture was there, that was what? Organic farming. So organic farming, what is one-liner meaning of organic farming? When we are not using any agrochemical, like chemical fertilizer, pesticide, such type of farming is called organic farming. So in organic farming, we are not using agrochemical. We are using manure in place of fertilizer. Got the idea? Understand the idea? Such farming is called organic farming. What is the merit and demerit associated with organic farming? Organic farming is best in all way. Only one bad things are there, demerit are there, that is less yield. Right? So initially organic farming going to conduct it all across the globe. But these organic farming not sufficient to meet the demand of the food of the growing population. So government of India, in all across the world people impose, but I am talking in Indian reference, government of India impose what? Green Revolution. They adopted Green Revolution. So what is Green Revolution? Very first question arises, and what changes occur with the Green Revolution? A question arises in the mind. Na? So Green Revolution is an initiative of government to increase the productivity of the food grain in the country. Question arises, what government did in Green Revolution? So productivity of the food grain increase in country. So government work in two areas. First area, improve seed quality. If better seed is there, productivity will also better. So government work in one area, what? Improve the seed quality. How government improve the seed quality? They give a lot of fund to Indian Agriculture Research Institute, ask scientists develop better variety of seed. In such a way, some semi-door variety came. Now, after second job, what government did in which one? Green Revolution. So they did agri management. Agri management. What is agri management here? So in agri management, government work in two areas. One area government work in improve the irrigation system. Water. What are one of the major reason of the death of the plant? Drought. So they develop the canal, they develop the tube well, and they use agrochemical. Agrochemical is of two category. One is chemical fertilizer. Another is pesticide. So what are the possible reason of less yield? Government try to subside all problem. If good seed is not there, productivity is not good, so they improve the seed. If water is not there, plant will die, so they arrange the water, irrigation system, canal system, tube well. Many plant die, not growth occur better because of soil is not fertile. They added chemical fertilizer to increase the fertility. Many times crop die due to which one? Attack of pest. So they spray the pesticide on the crop to prevent from the pest. So these Green Revolution definitely help us to increase the productivity of food grain in our country. Got the idea? Almost wheat production is going to increase, rice production increase. Getting the things or not? So they increase the productivity of the food grain. So what are the advantage we got? But Green Revolution shifted our agriculture from which one? Organic agriculture to agrochemical base. Agrochemical base. These are shifting occur during Green Revolution. So agrochemical base have only one merit, high yield, but everything bad. It is not sustainable, it is not cheaper, it is expensive, it is hazardous. Getting the thing. But one good thing in agrochemical based agriculture is what? Productivity is more. See, in India still now more than 90% agriculture is what? Agrochemical agriculture. Now we are going to talk about tissue culture. Got the idea? So then we will talk about role of biotechnology in agriculture. So we are going to talk about tissue culture. In tissue culture, what we are doing, so I am telling some of the terminology, meaning of some of the terminology, and then we will talk about here. So some of the terminology, first terminology is what is tissue culture. You must know. Second thing, what is micropropagation? Third one is what is totipotency. Fourth one is what is explant. Next is what is callus. Next is what is plantlet. Got the idea? Next is what is soma clone. These are the terminology you must know. I am telling what is tissue culture. So tissue culture is a technique of micropropagation. What is tissue culture? Technique of micropropagation. What we do in micropropagation? Producing thousands of plants from an explant during tissue culture is called micropropagation. What is totipotency? Potential ability of a plant cell to produce whole plant is called totipotency. What is explant? Any part of the plant we use during tissue culture for micropropagation is called explant. What is callus? Callus is undifferentiated mass of cell. What is plantlet? New plant produced through tissue culture is called what? Plantlet. What is soma clone? All the plant produced as a result of micropropagation are genetically identical and they are called soma clone. Got the idea? Now how we do the tissue culture? We are taking a culture. I'm changing the color. We are taking a culture medium. In culture medium, we are taking a which one? Nutrient solution. Nutrient solution is what? Sugar solution, source of carbon, sucrose. Vitamins we are taking. Minerals we are taking. Got the idea? Amino acid we are taking. We are taking plant growth regulator like which one? Auxin and cytokinin in culture medium. So these are the what basically explant. So in beginning we are keeping the auxin and cytokinin ratio equal. After some time explant is going to differentiate it for dividing and forming a ball like a structure, that is undifferentiated mass of cell. It is called callus. So callus for callus formation it is important auxin and cytokinin ratio are equal. Now what we are going for subculturing. We are shifting the callus into another culture medium. This time cytokinin is greater than auxin. It will differentiate the shoot. Next subculturing we are doing. Shifting the culture medium is called subculturing. This time auxin is greater than cytokinin. Shoot is already there. Who also develop? Root also develop. Root. So the new plant form is called which one? Plantlet. Now slowly we are exposing the external environment hardening. Because here chlorophyll is not there, because for chlorophyll synthesis light required. We are doing in lab. So slowly we are exposing in which one? Outer environment. So slowly chlorophyll synthesis occur. Got the idea? Later on they become compatible to grow in the outer environment and then we grow the plant. So we can make thousands of plants in very short span of time by a process of which one? Tissue culture. Now we are going to talk about somatic hybridization. Somatic hybridization, try to understand here, somatic hybridization. What we do in the somatic hybridization? Protoplast fusion. Protoplast fusion we are doing during the somatic hybridization. Getting the idea? Suppose that I am taking here a plant cell. Which plant cell? Potato cell. And I am taking here a tomato cell. It is a plant cell. They have the cell wall. It is which cell? Potato cell, and here it is tomato cell. They have the cell wall. So their cell will not fuse when cell wall is there. So when we are using enzyme, which one? Cellulase and pectinase. Cellulase plus pectinase. They digest the cell wall. After digestion of cell wall, who will remain? Protoplast. Here protoplasm is there and plasma membrane is there. This is called protoplast. Protoplast of potato cell, protoplast of tomato cell. Then we are fusing it. To increase the chances of fusion, we are giving which one? Source of energy, electric spark here. And plus we are using a chemical polyethylene glycol. It increases the chances of protoplast fusion. Both somatic cells fuse together. Got the idea? Now we are growing this one and making a plant. Now the plant name we have given which one? Pomato. What was our intention in agriculture? Ki in the same plant we got two crops. Our intention was ki from a same plant under the soil, potato grew, above the soil tomato came. But unfortunately, or NCERT wrote, unfortunately, all the desired combination of character not expressed. Not expressed. Got the idea? Presence of gene is a different thing, gene expression is a different thing. So unfortunately, all desired combination of gene not expressed. So this tomato is not available for commercial use. But how long it will not express? Scientists are working. A time will come now when we got all desired combination of result. So in coming future, it may possible na tomato in agriculture become a reality in their life. Farmer are growing. So how benefit will occur? In same time duration, in same cropland, we are getting how many crop? Two crop. So this is also used for which one? Improvement of which one? Crop. Got the idea? Somatic hybridization. Now we are going to talk about biotechnology. Which one? Agriculture by using GMO. Now we are going to talk about what? Agriculture by using genetically modified organism. So this is over. Now we are going to talk about agriculture by using genetically modified organism. Look, so with genetic modification, what we are doing? We are making crop more tolerant to abiotic stress. Right? Through genetic engineering, we are able to develop such type of plant who are more tolerant to drought, cold, salinity. Got the idea? So what happened? Plant survival chance will increase. Na? If plant survival chance will increase, what happened? Productivity increase, plant will not die. So biotechnology helping us to develop such kind of variety who have more tolerance to abiotic stress like cold, drought, salt and heat. Biotechnology also help us to reduce our reliance on chemical pesticide. So with the help of biotechnology, we developed many pest resistance plants like BT cotton, BT brinjal, BT corn, BT soybean, BT tomato, BT rice. These are BT. What the meaning of BT? Meaning of BT is what? They have a gene from Bacillus thuringiensis bacteria. And this gene is acting as what? Insecticidal. Understanding the idea? Clear in the mind or not? So when we, means through genetic engineering, we develop such type of plant who are pest resistance. Earlier pest was coming, killing the plant. Now when pest is coming, plant will kill them. Got the idea? So our dependency on chemical pesticide will decrease or not? Yes, of course. Now third, help to reduce post-harvesting loss. Beautiful example is flavor saver variety of tomato. Beautiful example is what? Flavor saver variety of tomato. What is the problem of tomato? Tomato have less shelf life. It is going to rotten after few days. When you go to market, you prefer to purchase a tight tomato rather than a loose tomato, because loose tomato will rotten very faster. So through genetic engineering, what we did, we make a genetic modification and we find that in tomato there is a gene is present. And these genes are releasing an enzyme that is what? Polygalactourinase. These enzymes digest cell wall. So what we did through the genetic engineering, we performed the silencing of this gene. Now who will not produce? Polygalactourinase will not produce, whose degradation becomes slow down. Enzyme speed up the degradation process, na cell wall degradation becomes slow down and tomato cell wall remain tighter for longer period. So who increase here? Shelf life is increased here. Now genetic modification increase efficiency of mineral usage by plant. Here you just require to understand. I am telling you example. What is the advantage of increase the efficiency of mineral usage by plant? So this prevent the mineral to exhaust early from the soil. How? You just try to understand with a car. A car is giving 20 kilometer average in 1 liter. One is giving 10 kilometer in 1 liter. Which car engine using fuel more efficiently? 20 kilometer. So who will save the fuel? 20 kilometer. In likewise, when efficacy of the mineral usage will increase, the requirement of mineral will decrease. Got the idea? So through genetic engineering, we able to develop like this. Now we enhance the nutritional value of the food through the genetic engineering. And beautiful example is what? Golden rice. In golden rice, what we did, we added 2 genes, one from the bacteria, another from the daffodil flower. And they are going to produce which one? Beta carotene. And when beta carotene is going to enter in our body, they form which one? Vitamin A. So golden rice is a beautiful example of it. Now we are going to talk about Bacillus Bt cotton. Actually, what is the problem of the cotton? What is the problem of the cotton? Cotton is a cash crop, and cotton get attacked by an insect pest called bollworm. Cotton get attacked by which one? Bollworm. Got the idea? Now what we did through the genetic engineering, what we did? We developed such a type of cotton plant who have gene for Bacillus thuringiensis bacteria. Now you can say, sir, why Bacillus thuringiensis is that much important? Let me say something about Bt. Actually, Bacillus thuringiensis is a soil bacteria. These Bacillus thuringiensis have a gene called Cry gene, CRY gene. These Cry gene release a crystalloid protein. These crystalloid protein released by Bacillus thuringiensis in certain stage of their life. And this protein is an insecticidal protein. When these protein enter in the midgut of the insect, at alkaline pH of the midgut, this protein, that is an insoluble crystalloid protein, becomes soluble and it bind with the midgut. It bind with the epithelium of the midgut and create pore in the midgut, and through that pore a lot of water enter and lysis of the insect occur, death of the insect occur. Are you getting the idea or not? Right? So that done by the crystalloid protein released by Cry gene, and these Cry gene present in which bacteria? Bacillus thuringiensis. But these not harm the bacteria. Why? Because these enzyme crystalloid protein present in bacteria in inactive form. It present as protoxin. Got the idea? It is insoluble form it is present. It is not present as a toxic form there. But when it becomes toxic at alkaline pH of the midgut, it becomes toxic. But these Bt not kill all the insect. Got the idea? It is killing whom? Got the idea? It is killing whom? So NCERT wrote here, this crystalloid protein, I am just coming on that particular line. So NCERT wrote, some strain of Bacillus thuringiensis produce protein that kills certain insects such as Lepidopteran insect. Lepidoptera is the order of class Insecta, and it includes tobacco birdworm, armyworm, or even bollworm. Bollworm is also which one? Lepidopterans. Got the idea? It is also killing Coleopteran insect. Who included here? Beetles. Don't forget this line. And it also kills dipteran insect, flies and mosquito. Is dipterans. So it is not killing all the insect. It is killing how many order of insect? 3 order of insect. Diptera, Coleoptera, and which one? Lepidoptera. Understanding the idea? Insect have total 29 order. Out of 29 order, it is killing how many order? 3 order of the insect only. Are you getting the idea? Is it clear in the mind? Now, what is the storyline? A storyline is this. You try to understand. A storyline is this. This is the Cry gene. Cry gene. We need a which one? Vector DNA. Suppose that this is Ti plasmid. Suitable vector for plant is which one? Ti plasmid. We cut both with the same enzyme and join with the help of which one? DNA ligase. Now I am taking here a which one? Tobacco plant cell. Introducing this one. Now by tissue culture, we are making thousands or lakhs of which one? Genetically modified tobacco plant cell. It is called which one? Sorry, tobacco I am telling. Cotton plant cell. Cotton plant cell, not tobacco. Mine came on RNA. I got the idea. Cotton plant cell. Now, by tissue culture, we are making which one? Bt cotton. Now, every cell of the cotton have which one? These chrysin. In every cell, who is there? Crystal white protein. Now, who is coming to feed? Bulbworm coming to feed. That plant cell enter into which one? Their food pipe. And in food pipe, in midgut, what happen? In gizzard, churning of cell occur. Protein will release, they will enter in midgut, and in midgut it becomes soluble at alkaline pH. Alkaline pH is important. Alkaline pH will important. At alkaline pH, protoxin converted into which one? Toxin. And when it converted into toxin, it bind with the midgut, create pore, right? Through pore, water will enter. What happened? It will swell and death occur. Now you don't forget please the two names, Cry1AC, Cry2AB is gene for which one? Cotton bollworm. And another gene which one? Cry1AB provide resistivity against corn borer. So who is over? Bt cotton is over. Are you getting the idea? Now we are going to talk about RNAi. I am telling 5-6 questions from here. All RNAi will over. I am telling 5-6 questions. Nothing will come in exam more than that. Listen it carefully. Here also people have a lot of apprehension and tension in RNAi. So in revision, this revision program, I am not talking all this nonsense like Dicer, Ergonaut, RISC, these things I am not going to introduce. So I am writing some question. If you are able to handle it, RNA is handled. First, what is RNAi? Next question. So answer of this question is what? RNAi is a method of cellular defense in all eukaryote, not in prokaryote. It is a method of cellular defense in all eukaryote. Question arises how? How it performs the cellular defense? Answer, by post-transcriptional gene silencing. By post-transcriptional gene silencing. So RNA is a method of cellular defense. How? By post-transcriptional gene silencing. Got the idea? So another question. Nematode infect. Answer of the question is plants, fungi, animals, and even human. Next question, AMI infect. AMI is what? Myelogyne incognitia. Infect, answer is what? Root of tobacco plant. Root of tobacco plant and decrease their yield. Decrease their yield. MI infect which one? Root of tobacco plant and decrease their productivity. So this is the problem. So what we did in RNAi, actually it is developed by a scientist whose name was Andrew Fire and C. Mello. What they did, by genetic modification, he made such kind of tobacco plant that now When mi is coming, who is going to die? Mi is going to die. Earlier, who is dying? Tobacco is dying. After genetic modification, who is dying? Mi is dying. Now I am asking another question. What is the source of nematode specific gene? What is the source of nematode specific gene during RNAi? Tell me. So answer of the question is what basically? RNA virus. Second answer is what basically? Transposon. Transposon is which one? Jumping gene, it is a mobile genetic element. Another is which one? RNA virus, which replicate via RNA intermediate. Now next question is, vector used during RNAi. Simply answer is which one? Agrobacterium. Agrobacterium. Next question, how we introduce nematode specific gene in host cell? Answer, in such a way it produce dsRNA, double stranded RNA. Out of double stranded RNA, it is a dsRNA. It is 5' to 3', 3' to 5', 5' to 3' is called sense strand, and 3' to 5' is called antisense strand. Now what NCERT wrote? NCERT closing the chapter now. NCERT wrote कि when a nematode feed, and even they not wrote in that clear language, what happened? These dsRNA from tobacco plant enter in the food pipe of which one? Nematode. It is I am telling here, not even NCERT wrote. Now from there these dsRNA enter into which one? Nematode cell. And in nematode cell what it will do? It is a vital gene of nematode, and this is the mRNA of the nematode. And these dsRNA, even NCERT wrote dsRNA, not dsRNA. It is which one? Antisense strand. It is also called guide strand. It pair with the mRNA and stop the expression of these mRNA. Protein will not synthesize, and these cell will die. Like many cell will die, nematode will die. Are you getting the idea? So that is the storyline of which one? RNAi. So these are the questionnaire. You read after that NCERT. Every possible question is answered here. I am not going in the mechanism part here because it is not in NCERT, and even it never asked in exam. So examiner simply asks the question like, what is the vector used? Agrobacterium. What is the source of nematode specific gene? RNA virus or transposon. How we introduce nematode specific gene in such a way it produce double stranded mRNA. These double stranded mRNA, which part especially? Antisense part join with mRNA of the nematode cell, and they stop their expression. How they enter in nematode body? When they are eating the plant cell, from plant cell first mRNA enter in which one? Food pipe. From there absorbed by cell, and then they bind with which one? Nematode mRNA, and stop their expression. Are you getting the idea? So these are the probable and potential questions from RNAi. RNAi is over. Got the idea? Right? Clear? Once again, see what is RNAi? Method of cellular defense. In RNAi, I stand for interference. How we to do the cellular defense? By post-transcriptional gene silencing. Nematode infect plant also, fungus also, animal also, human also. Mi infect root of tobacco plant. What is the source of nematode specific gene in RNAi? RNA virus or transposon. Vector use is what? Agrobacterium. How we introduce the nematode specific gene into host cell? In such a way it produce double stranded RNA. Out of two strand, one is sense, another is antisense. These antisense will pair with the mRNA of the nematode and stop their expression. Due to that reason, who die? Nematode cell will die, and if many such cell will die, nematode will die. You got the idea? So this is about the, now Other thing is time pass here. You read it. It is not my job. Only one thing I am telling you is GEAC. Every alternate year this question came. Genetic Engineering Approval Committee. In India, it look after safety associated with GM research, and it also associated with giving licensing to which one? GM research and GM product. Got the idea? Genetic Engineering Approval and Appraisal Committee. So both chapter is over, biotechnology is over. Now in another 5 minutes later, I will start 5 or 7 minutes later. I will start the molecular basis of inheritance. Getting the idea? Is it clear? Then principle of inheritance, then reproduction. Okay, right? So biotech over. So 2 hour, 2 chapter, 3 more hour is there. So we will just try to maintain somehow more speeds in these coming chapters. Done, over, right, Ji. So it's a break of 5 minutes. Then we will once again came. Okay, Ji, thank you very much. So see you just after 5 minutes. No, it's okay? No. So, is it okay now? Anything. So now look, let's start. So what I am telling, we are starting with the new chapter, that is molecular basis of inheritance. See the sequence of the event for the genetic material. Mendel told factor is responsible for character. You already studied earlier in principle of inheritance. And Mendel told factor is something. Right? Later on, Sutton and Boveri came and he told what? Chromosomal theory of inheritance. And when we studied chromosome, we find in chromosome who is there? DNA is there, RNA is there, histone protein is there, non-histone protein is there. So who is the act as genetic material? So NCERT wrote further that who is a putative genetic material? DNA. So if we talk about DNA act as genetic material in almost organism. Exceptions are there, that is some virus. Exception is what? Some virus where RNA is genetic material. Where RNA act as genetic material. So in most of the organism, who is the genetic material? DNA is a genetic material. In some viruses, who is the genetic material? RNA is the genetic material. So RNA, what RNA is serving? So I'm writing here, RNA main function is what? It acts as which one? Messenger. So RNA act as messenger RNA. RNA also act as adapter. Who is acting as adapter? tRNA. RNA also have catalytic and structural role. And a structural role. And who is this? Ribosomal RNA. Ribosomal RNA act as catalyst also, ribozyme, and ribosomal RNA is a part of ribosome, a structural role also. Are you getting the idea? So this page is almost over. Got the things? Now we are moving ahead. Now we are going to talk about which one? DNA. So some data is important here. Please don't forget. From here, questions is going to ask in the Neet exam. If you see the P by Q, also question is there, is available from there. So what they are writing, the length of DNA is variable in different species, because length of DNA depend upon base pair. Length of DNA depend on base pair. So number of base pair is variable in different species, or you can say number of nucleotide is variable in different species. So what data NCERT give for the proof of it? They told, just look, a bacteriophage is there known as 5x174. There how many nucleotide is there? 5386 nucleotide is there. Here base pair is not mentioned, be careful. Lambda phage bacteriophage have a 48502 base pair, you know about it. Then NCERT wrote Escherichia coli have 4.26 into 10 to power 6 base pair. And in human haploid cell or human genome, how much base pair is there? 3.3 into 10 to power 9 base pair is there. Got the idea? So what is DNA basically? DNA is a which one? DNA is a which one? It is a heteropolymer. Heteropolymer of nucleotide. Heteropolymer of nucleotide. 4 type of nucleotide are present in which one? DNA. Are you getting the idea? Then DNA talk about the structure of the polynucleotide. So see what DNA talk about the structure of the polynucleotide. I am explaining the structure of the DNA, sorry, NCRT only, diagram of the NCRT only. Have a look here. So look here. So here it is what basically? It is a polynucleotide chain. Why polynucleotide chain? How many nucleotide is there? 1 nucleotide, 2 nucleotide, 3 nucleotide, 4 nucleotide. So in one nucleotide, how many component is there? In one nucleotide, this is nitrogenous base. It is which one? Pentose sugar. And this is what basically phosphate. So one nucleotide consists of nitrogenous base, right in short, nitrogenous base plus pentose sugar plus phosphate. Now in a nucleotide, nitrogenous base join with pentose sugar by N-glycosidic bond. N-glycosidic bond. And in formation of N-glycosidic bond, NCRT wrote that pentose sugar always participate with its first carbon. And when pentose sugar join with which one? Phosphate, it is joined with which one? Ester bond. Ester bond. So between sugar and phosphate, ester bond is there. Between sugar and phosphate, which bond is there? Ester bond is there. And between nitrogenous base and sugar, which bond is there? N-glycosidic bond is there. N-glycosidic bond is present. Got the idea? Is it clear in the mind? So here it is a nucleotide. In a nucleotide, how many type of bond is there? N-glycosidic bond and ester bond is there. Now question arises, how 2 nucleotide is joining? Look here. Here it is 2 nucleotide. From here to here, how many nucleotide is there? 2 nucleotide is there. So phosphate join, here one phosphate join with which one? Both sides. Nah? So here it is one ester bond. And this side also another ester bond. So 2 nucleotide of the same strand join together by which bond? 2 nucleotide of, it is very important line, 2 nucleotide of same strand join together by 3' to 5' phosphodiester bond. Phosphodiester bond. 2 nucleotide of the same strand joined together by which one? 3' to 5' phosphodiester bond. Now who is 5' here? How to know in a strand of the DNA who is 5' and who is 3'? So look here. Here it is 5th carbon and phosphate attached with the 5th carbon. So it is called 5'. So and tricks where phosphate is present in the last. Free phosphate, that is called 5'. Here it is sugar. Sugar have OH group at which position? 3rd position. So it is 3'. So DNA have how many strands? 2 strands. Always we write like na 5' to 3', 3' to 5'. Both the strands are what basically anti-parallel. 5' free phosphate, 3' at sugar at 3rd position, 3rd carbon, free hydroxy group is there. So it is called which one? 3'. Are you getting the idea? So if we talk about the structure of a polynucleotide. Picture came in mind or not? If you want to draw it, you can draw like this. Have a look here. This is phosphate. This is sugar. This is nitrogenous base. One nucleotide. Another phosphate. Another sugar. Another nitrogenous base. Another phosphate. Another sugar. Another nitrogenous base. Are you getting the idea? So in such a way, here phosphate is there, so it is 5 prime. Here OH present on third position, so it is 3 prime. So this is the structure. This is the structure of a which one? Polynucleotide. Are you getting the idea? Understanding the idea? So polarity 3 and 5 prime came in the mind. Is it all right? Is it clear? Is it done? Right. Now we are moving ahead. Now if I ask a question, small question to you, what is the basic difference between DNA and RNA? I think since childhood everyone is studying, so you also have to keep these things in mind. DNA, RNA. So here usually which one? Double stranded. Usually it is which one? Single stranded. Exceptions may possible. Got the idea? Later on DNA act as genetic material in most organism and act as genetic material in few virus. Genetic material in few virus. Another difference between DNA and RNA, it have deoxyribose sugar. Here it have ribose sugar. What is the difference between deoxyribose and ribose sugar? In deoxyribose sugar at second position, second carbon, hydrogen is present. In ribose sugar, who is there? OH is present. If you make a diagram like this, I am not emphasizing everything. I am emphasizing which one? Second carbon. Here OH group is present in ribose sugar. Got the idea? And in deoxyribose sugar, only who is present at second position? At second position, who is present? At second position, hydrogen is present. So this is in deoxyribose sugar, and this is in which one? Ribose sugar. Are you getting the idea? Remaining are same. Is it clear? Later on here, who is present? Thymine is present. Here it is uracil is present. So these are the structural differences in the DNA and RNA. Got the idea? Right? Over. Now if we talk about I am asking a question here. I am asking a question. What is this? C. Cytosine. What is this? Cytidine. What is this? Cytidine monophosphate. PHAT. Cytidine monophosphate. Cytosine is a nitrogenous base. Cytidine is a nucleoside. And cytidine monophosphate is a nucleotide. Or other name of cytidine monophosphate is cytidyllic acid. Got the idea? Like I am writing adenine, nitrogenous base, adenosine, nucleoside, adenylic acid, or AMP, ADP, ATP, these all are what basically? Nucleotide. Number of phosphate in a nucleotide may be 1, 2 or 3. Got the idea? So adenosine monophosphate is other name of adenylic acid. But ADP and ATP is also what basically nucleotide. Got the idea? What is the difference between nucleotide of DNA and RNA? So if it is written deoxyadenosine, deoxyadenylic acid, deoxyadenylic acid, if it is written, it is present in which one? In DNA. If simply written adenylic acid, it is present in which one? RNA. Because deoxyribose sugar is present, na? Likewise, we will write the name of everyone. Got the idea? So there is no time to discuss on these more. I am asking a simple question. You tell me. What is uridine? What is uridine? Uridine is a nitrogenous base, nucleoside, nucleotide. What is this? Answer it. What is uridine? It is a nucleoside. Got the idea? Now we are moving ahead. Now, few important discovery you please don't forget. That is also important to keep in mind. First thing you have to know it that DNA is an acidic substance present in nucleus. First identified by Frederick Miescher. In which year? 1869, and he given the name which one? Nuclein. So term nuclein is given by Frederick Miescher, and DNA is an acidic substance present in nucleus is told by which one? Frederick Miescher. Got the idea? Now, double-stranded helically coiled structure of DNA is given by Watson and Crick. Is given by whom? Watson and Crick. Their model is based on X-ray data diffraction result proposed by Wilkins and Franklin. Got the idea? So model, double standard model of the DNA is based on X-ray data diffraction result of which one? Franklin and Wilkins. Got the idea? Now further it is written, na? If we talk about NCERT wrote double helix model of the structure of DNA, it is proposed. One of the hallmark of their proposition was base pairing. What is the hallmark of the proposition? Base pairing. What base pairing? Adenine always pair with thymine, cytosine always pair with which one? Guanine. Adenine with thymine with double hydrogen bond and cytosine with guanine with triple hydrogen bond. So these proposition is also based on the idea of Irving Chargaff. What Irving Chargaff told? In a double stranded DNA, Chargaff, we also popularly call Chargaff rule. So what is this? In a double stranded DNA, it is applicable for double stranded structure. So in a double stranded DNA, adenine is equal to thymine, cytosine is equal to guanine. It is equal to. So in a double stranded DNA, what Chargaff told? Adenine is equal to thymine and cytosine is equal to guanine. And that is the reason NCERT wrote that the hallmark of the proposition of the Watson and Crick model is what? Complementary base pairing. Adenine pair with thymine always they told. Cytosine pair with guanine is also told. And further it is wrote this proposition was based on Chargaff finding. What Chargaff find? Irving Chargaff. In a DNA, adenine is always equal to thymine. Why it is always equal to thymine? Because adenine always pair with thymine. Cytosine always equal to guanine. Why it is always equal to guanine? Because cytosine always pair with the guanine. So we can say like na in a Chargaff rule, purine is equal to pyrimidine. You see na, always adenine, purine, thymine, pyrimidine, cytosine, pyrimidine, guanine, purine. Always a purine pair with pyrimidine. So number of purine and pyrimidine is equal. So we can write A plus G upon plus C plus T equal to C plus T, and we can write like A plus G upon C plus T is equal to 1. This is Chargaff rule. This is Chargaff rule. Who is variable from a species to a species? A upon T, A plus T upon C plus G is not equal to 1. It is constant for a species. A species and variable from a species to a species. And variable from a species to species. Got the idea? So this is about the history associated with how many scientists name you must not forget. Frederick Misser, he told nuclein. Watson and Crick, he have proposed a famous double-stranded helically coiled structure of the DNA. Franklin and Wilkins, he give the X-ray data diffraction result of the DNA. Based on that, model of the DNA is based. Next, Chargaff told what? Irving Chargaff told in a DNA, adenine is always equal to 1. Sorry, adenine always equal to thymine and cytosine is always equal to guanine. Got the idea? Is it clear? So in a DNA, according to Chargaff, A upon T equal to 1, C upon G equal to 1, according to Chargaff. Got the idea? Is it clear? Now we are going to talk about one question, not more, one question based on which one? Chargaff rule. So because numerical nature of question going to ask from here, so wherever some of the student is stuck in, it is our moral duty to address it. So I am asking a question based on Chargaff rule. Okay, Ji? Is it done? Clear? Huh. A DNA have just a minute. Which is 68 nanometer long, have 20% adenine. Then find out the answer of following question. From here question is going to ask, that is the reason I am addressing it. 68 nanometer long is there. Got the idea? And 20% adenine there. They find the answer of following question. First, number of base pair. Second question, number of glycosidic bond. Third question, number of phosphodiester bond. Next question, number of nucleotide of cytosine. Next question, number of hydrogen bond. So all possible questions I wrote here. Can you solve it? Help me to solve it. So I am giving the question which one? Length of the DNA is how much? 68 nanometer. Now tell me the base pair. Tell me the base pair. How many base pair will come? You please answer first. How many base pair will come? 68 nanometer is there. How many base pair will come? Who are online, please at least answer. Who are offline, of course, I can't connect with them. Who are online, answer it. 68 nanometer is there. So distance between two base pair, you better know this basic idea, you know, distance between two step or two base pair is how much? 0.34 nanometer. So number of base pair equal to what will happen? 68 divided by 0.34. How much coming? Tell me. How much coming? 32 2 64. How much it will come? 200 base pair come na? 200 base pair came. Okay. So first answer is over. First answer is over. Number of base pair came. Number of glycosidic bond. You don't forget glycosidic bond is just double of which one? Base pair. So glycosidic bond equal to what basically? Double of base pair. So how many base pair is there? 200. So number of glycosidic bond equal to 2 into 200 equal to how much? 400. Number of phosphodiester bond. Third question. Everybody know what is the formula is there? 2n minus 2. 2n equal to base pair. So 2 into 200 minus 2. 400 minus 2 equal to how much? 398. Did you got the idea? Number of nucleotide of cytosine. Total nucleotide is how much? Total nucleotide is double of base pair. Double of base pair. So base pair is 200. So 2 into 200 equal to 400 nucleotide is there. 400 nucleotide is there. And how much percent is adenine? Adenine is 20%. So thymine also 20%. Total 40%. 600 minus 40 equal to 60%. 60% have two partner. 30% cytosine, 30% guanine. Did you got the idea? 30% cytosine, 30% guanine. I am asking nucleotide of cytosine. So how much nucleotide is there? 400 into 30 upon 100. 0, 0, 0, 0. How much? 120. Are you getting the idea? Understanding the idea? Later on I am asking what? Number of H bond. So adenine, thymine together how much percent? Adenine, thymine is how much percent? 40%. 40% of 200. Cytosine, guanine, triple bond is how much? 60% of 200. What should I write? 40 upon 100 into 200. Here what should I write? 60 upon 100 into 200. How much it is coming? 80. How much it is coming? 120. Right? So adenine, thymine 80. Adenine, thymine, how much is there? 80. 80 into 2. Why 2? Double hydrogen bond, 160. Cytosine, guanine is how much? 120 base pair. 120 into 3. Why triple bond is there? How much? 360. How much it will come? 0, 520. Understand? Clear? Is it done? Is it clear? Over? Done? Understanding came? Every question is solved? Understood? Clear? Can I move ahead? So these are the questions based on Chargaff rule I posed and the DNA length based question. Getting the things or not? Now we are moving ahead. Give your attention. Now, if we talk about the structure of DNA, just have a look here. So, if we talk about the structure of DNA, it is a double helically coiled structure like this. Are you getting the idea? It is a double stranded helically coiled structure. From here to here, cancel. From here to here. It is called one turn because from or pitch of the DNA, one turn, or it is also called pitch of DNA. Pitch of DNA. Got the idea? In one turn have how much? 10 base pair. Length is how much? Length is how much? 3.4 nanometer. Distance between two steps is how much? 0.34 nanometer. Maximum distance between two strands is how much? 2 nanometer. Axial rise is how much? 36 degree. Because total 360 degree. So angle between two steps is how much? 36 degree. So these are the data, please you keep in mind. One turn of pitch equal to 10 base pair. Length of a pitch 3.4 nanometer. Diameter 2 nanometer. Distance between two steps 0.34. And what the idea? Axial rise 36 degree. It is which structure of DNA? It is secondary structure of DNA. It is which type of helix? It is right-handed helix. It is right-handed helix of the DNA. Are you getting the idea? It is right-handed helix of the DNA. Getting the idea? Is it clear in the mind? It is a secondary structure. It is a right-handed helix. It is a which form of the DNA? B form of the DNA. Got the idea? So this is about the structure of a DNA, double helically coiled structure of the DNA. Another structure of the DNA showing in the diagram here, have a look. So DNA is what basically made up of two strands and many steps. Both strands are anti-parallel to each other. It is 3' to 5'. It is 5' to 3'. Both the strands are anti-parallel to each other. So these strands, sugar and phosphate in the strand making the backbone of the DNA. And here nitrogenous base pair making the steps of the DNA. So strand is made up of alternate arrangement of phosphate, sugar, phosphate, sugar, phosphate, sugar. And steps are made up of nitrogenous base pair. In steps, always what happen? Adenine pair with thymine with double hydrogen bond, cytosine pair with guanine with triple hydrogen bond, and nitrogenous base pair is project inward to the which one? Strand. It is a strand and toward inward which projected? Nitrogenous base is projected. So this is the simplified structure of DNA. This is the double helical structure of DNA. Now we are going to discuss about central dogma of molecular biology. It is proposed by whom? Examiner will ask. So Francis Crick. Francis Crick proposed the central dogma of which one? Molecular biology. What is saying? What central dogma is saying? Central dogma is saying about flow of genetic information. How genetic information is flowing? Information from DNA travel to RNA and from RNA it travel to protein. So if we talk about DNA to RNA formation, mRNA formation is what basically here transcription, and mRNA to protein synthesis is called translation. And DNA to DNA synthesis is called replication. In some viruses, like retrovirus, mRNA convert into DNA, it is called what? Reverse transcription. It is called what? Reverse transcription. It is discovered by Temin and Baltimore, so it is also called Teminism. It is also called Teminism. These reverse transcription example you can say about which one? HIV. Enzyme involved here is which one? Reverse transcriptase. Reverse transcriptase. It is a RNA dependent DNA polymerase. You got the idea? So this is about the central dogma of molecular biology, flow of genetic information. Later we are moving ahead. Give your attention. Now we are going to talk about packaging of DNA. What you must not forget from here is, actually, reading NCERT taking more time. It is more better without NCERT observing, you just say. It become more faster. So you just try to understand about packaging of DNA. Problem is what? Problem is your nucleus is 10 to power nucleus minus 6 meter diameter. And DNA present in human cell, an example I am telling is 2.2 meter long. So for this 2.2 meter is the length of one DNA or all 46 DNA. When you are placing all 46 DNA present in a human diploid cell side to side, got the idea? Side to side, one after another, the total length coming 2.2 meter. So 2.2 meter is not the length of one DNA, it is the length of all 46 DNA present in human cell. You got the idea? And how it placed? One after other. One DNA, another DNA, another DNA, another DNA, and all 46 DNA, it will come how much? 2.2 meter. You got the idea? Is it clear in the mind? Right? So 2.2 meter is the length of all 46 DNA. Is it done? Clear? Can we move ahead? So problem is what? 2.2 meter long DNA, how it will accommodate into which one? 10 to power minus 6 meter diameter structure. So it is going for a very precise folding. So how the folding of the DNA occur, that we are going to discuss about through which one? Nucleosome model. So what NCERT is saying? NCERT is saying what happened basically, DNA, which is a negatively charged structure, DNA is which one? Negatively charged. It going to wrap around a histone protein octamer. It is going to wrap around which one? Histone protein octamer. This histone protein octamer is made up of 4 type of protein. H2A plus H2B plus H3 plus H4. These are the 4 type of protein present in this ball-like structure, histone protein octamer. And everyone have how many copy? 2 copy. So 8 small ball joined together and making this larger ball, which is looking green color here. Got the idea? So how many protein is there? 4 type of protein is there, and there are 2 number of each. And these protein are rich in amino acid, lysine and arginine. And you better know it is a basic amino acid. So basic amino acid, due to that reason, these histone protein octamer are rich in basic amino acid. So overall charge developed on the octamer is which one? Positively charged. And DNA is negatively charged. Why DNA is negatively charged? Due to presence of phosphoric acid. So what happened? DNA wrapped around histone protein octamer. I'm just giving you a concise diagram. So I'm just making diagram here only, so you will understand it better. So I'm just making here, like suppose that this is a ball, another ball, another ball. It is all histone protein octamer. You just try to understand this is all histone protein octamer. I'm going for a simply representative diagram. So DNA, I'm just making it is DNA wrapping here. So when DNA wrapped around histone protein octamer, it is making a structure called chromatin. It is making a structure chromatin. And one ball with DNA, wrapped DNA is called what basically? Nucleosome. So this is a nucleosome. DNA wrapped around histone protein octamer is called nucleosome. And what is this? This is called linker protein. Linker protein. And the name of linker protein is which one? H1. H1 is a linker protein. Got the idea? Now, how many base pair of DNA wrapped around the histone protein octamer? 200 base pair of DNA wrapped around histone protein octamer. So how it is wrapping? It is wrapping like this. It is wrapping like this. Later on, it is going for foldings. It is going for folding. When it is wrapped around, it is going to make a fold. Fold after fold. And it is going for 6-time folding, and it is going to make a solenoid structure. Got the idea? And later on, these chromatin, after folding, chromatin fiber will form, and later on it is going for further condensation. It is going for further condensation. And after further condensation, it is going to make a structure called chromosome. This is a chromosome. This is a chromosome. So how it is folding? So a DNA, earlier it is a DNA. When they wrapped around which one? Histone protein octamer, it is forming a which one? Chromatin. Got the idea? And when this chromatin is going for further condensation with the help of which protein? Non-histone protein. Further packaging when occur, it is going to form a structure chromosome. This is a chromosome of which stage? Monad stage. It is a chromosome with single chromatid. So one chromosome have how many DNA? One DNA present in a chromosome. And that's the reason NCERT told that nucleosome form the repeating unit of a structure called chromatin. Many nucleosome all together forming which one? Chromatin. Understanding the idea? You make it wrap here also. Many nucleosome forming a which one? Chromatin. So here what is this? This is the structure of which one? Bead on a string structure. So every bead is, this beaded structure is called what? Nucleosome. Nucleosome, and the overall structure you are finding here is what? Basically chromatin. Got the idea? Two type of chromatin is there, euchromatin and heterochromatin. Euchromatin is lightly strand, heterochromatin is darkly strand. Euchromatin is transcriptionally active, heterochromatin is transcriptionally inactive. Euchromatin is loosely condensed, heterochromatin is densely packed, loosely packed and densely packed. Euchromatin replicate in early S phase, heterochromatin replicate in less S phase. Are you getting the idea? So this is about the packaging. Got the idea? Now, how many nucleosome is present in human genome? It is too easy question, na? Human or human diploid cell. Human diploid cell have how many nucleosome? Human diploid cell have how many nucleosome? So human diploid cell have how many base pair? Human diploid cell have how many base pair? 6.6 into 10 to power 9 base pair. And in one nucleosome have how many base pair? 200 base pair. Divide by 200. 9 becomes 7. 3.3 into 10 to power 7 nucleosome is present in which one? Human diploid cell. Are you getting the idea? Now question arises, in eukaryote we have seen, is the genetic material in prokaryote is scattered? No. Even is in prokaryotes genetic material is not scattered. Now you can ask a question, sir, histone protein is not there in prokaryote, na? If it is not there in prokaryote, how it is arranged? So NCERT wrote, na, in prokaryote what happened? In prokaryote what happened? DNA is assembled with which one? A positively charged protein and forming a large loop of a structure called nucleoid. So even DNA is not scattered in prokaryote, it is forming a large loop of circular structure called nucleoid. How? By assembling along with which one? Positively charged protein. So this is about the euchromatin and heterochromatin. Now we are going to talk about the search of genetic material. Search of genetic material. I think rather than this, I will say more faster. So Wait a minute. Search of genetic material. Three important experiment is there. One experiment performed by which one? Griffith. And Griffith performed the experiment in 1928. What he did in the experiment, he had taken two different strain of pneumonia causing bacteria. One is which one? R strain. Another we have taken S strain. Got the idea? R strain is a virulent strain and S strain is a virulent strain. Got the idea? So these are two strain of which one? A Streptococcus pneumoniae bacteria. It is pneumonia causing bacteria. So what he have did, he have taken a mice in very first experiment and he inject live R strain. So mice will survive or die? Mice will survive because it is avirulent. In second experiment, he have taken mice, he inject live S strain, smooth strain. It is virulent, it causes pneumonia and death of mice will occur. In third experiment, he have taken mice and inject heat killed S strain. Bacteria they killed by heating first, then they inject. It is surviving because who is already dead? Bacteria is already dead. So how it did dead bacteria cause pneumonia? Surviving. Fourth experiment, what he did, he have taken a mice and inject live R plus heat killed S. Alone no one kill, but here they find death of the mice occur. When he did the post-mortem of the dead mice, he find after post-mortem or autopsy, he find live S is present. Question arises, he have taken heat killed S, how live S came? So he make a conclusion and that conclusion is called transforming principle. It is called transforming principle. What he told in the transforming principle, what conclusion he have given, he told कि actually what is the difference between S and R, I am telling you. In S strain, who is there? Polysaccharide coating is there. Or mucus coating. Or it is called which one? You can say like slime layer of capsule. So capsule is present in S strain. It is capsulated, R strain is non-capsulated. So here it is which one? Heat killed S strain and here live strain, live R strain. It is non-capsulated, it is capsulated. What he told, कि when we inject the mixture of both, what happens? Some material transfer from here to here. Heat killed S to live R. So who changed here? Genotype change, na? And these material who transfer make the polysaccharide coating here, mucus coating here. So when mucus coating came, it also become which one? Live S. And live S is virulent and kill the mice. Means whoever the material transfer from dead bacteria to living bacteria, heat killed S to live R have power to change the phenotype. And what phenotypic changes it did? Convert live R into which one? Live S. And it is called what basically? Transforming principle by Griffith. Got the idea? So this is about the Griffith experiment. Now, what we are going to deal, we are going to talk about the second experiment that is performed from 1933 to 44, and it is performed by which one? MacLeod, McCarty and Avery. Got the idea? And they told about which one? Biochemical character of the transforming principle. Got the idea? They did what basically? Biochemical character of the transforming principle. Who proved it? Who proved it? Avery, MacLeod and McCarty. Isn't it? Now what they did, how they proved, I am just going to tell you. I am just going to tell you how they did it and how they proved it. Isn't it clear? Now we are moving ahead. So they have taken which one? Heat killed S bacteria. And what they did, isolated the protein from this bacteria, isolated the RNA from this bacteria, isolated the DNA from this bacteria. And then they have taken which one? Live R bacteria. Live R bacteria. Live R bacteria. Live R bacteria. I am writing all together live R. Fight is what? Who is causing the transformation? So they were smart people. They know na कि भाई chromosomal theory of inheritance. Sutton and Boveri told character transferred by chromosome, but chromosome have DNA, RNA and protein, so any one will be the responsible for transformation. So from dead bacteria, they separate all these three. First they add protein in this. They find no transformation occur. Heat kill remain, live R remain live R. Then they add RNA in this culture medium. They find no transformation occur. And then they add DNA in this culture medium. This converted into which one? Live S. So what is proved? What is proved? DNA is transforming material. What is not known to Griffith, they told na DNA is transforming material or genetic material. Genetic material. Because it is changing the phenotype. But every people not get agree with them. And he proved it in a two different ways. One method of proof is this, another method of proof is what? They have proved it by another method also. What is the another method they used to prove it? Let me tell you another method he used to prove it. They have taken here which one? Heat killed. Here it is heat killed S side. And here it is live R side. Here it is live R side. So here they add enzyme protease. Protease will digest everything other than digest protein. No protein is there. After digesting the protein, they adding it here. Who converted? Live R converted into live S. Means who don't have a transformation? Protein, because here protein is digested. Color I am changing. Then they have taken next time. Here bacteria, heat killed. Here it is live R. So he add here RNase. When he add RNA, who is not there? RNA is not there. And then they transfer here. Who converted? Live S is converted. Means without RNA, live R converted into live S. Means who don't have a role? RNA don't have a role. Then the next he have taken here heat killed. Here which one? Live R. Here he added DNA. Mean they digested the DNA. When they are adding here, it is not converting. Means without DNA, these bacteria, when introduced into this bacteria, transformation not occur. So it is double proved what? DNA is a transforming principle, getting the things or not, transforming material by these scientists. Are you getting the idea? Understanding the idea? Clear or not? Right? Now we are going to move ahead. NCERT asked a question here. I hope you are aware about the answer of it. Okay, what is the difference between DNase and DNase? DNase is a which one? Enzyme, and it is a plural form of the DNA. Are you getting the idea? Understanding the idea? Got the idea? Is it? And also don't forget this line. Also don't forget this line. in these experiments. What NCERT wrote? Prior to work of Oswald Avery and co-worker, the genetic material was thought to be protein. But they break this myth and told who is the genetic material? DNA is the genetic material. Later on the last experiment, that is of which one? Hershey and Chase experiment. Give your attention. Hershey and Chase. I am taking diagrams, so no need to draw the diagram. Have a look here. This is Hershey and Chase experiment. Just try to understand. This is Hershey and Chase experiment. Actually, people was fighting about what? Who is going to next generation, DNA or protein? Now, unequivocal proof of it is given by Hershey and Chase. What Hershey and Chase have taken? They have taken a bacteriophage. Two set of experiment they did. One is this. In one set of experiment, they radio level the S35, protein by S35. Sulfur is present in protein, na? So he have taken the S35 isotopes, not S32. And in another set of experiment, they have taken P32. Phosphorus is present in DNA. Sulfur is present in protein. So they have radio level the odd element, one element of DNA, another element of protein. Got the idea? Now, when he infect with the bacteriophage to a E. coli bacteria who have radio level sulfur in it, after infection, blending and centrifugation, what they are finding? No radioactive S35 detected in the cell. In bacterial cell, who is not there? S35 not came. It means who not enter inside the bacterial cell. Protein not enter inside the bacterial cell. Protein not, see na, here it is. This is radio level. Who is not entering in the bacterial cell? Protein is not entering in the bacterial cell. If protein enter inside the bacterial cell, who must be there? S35 must be there, na? Got the idea? And when he radio level with the P32, what he find? Radioactive P32 is detected in bacterial cell. Means who enter in the bacterial cell? DNA enter or protein enter? P32 enter means DNA enter. If a protein cannot enter in the bacterial cell, how it will infect the bacteria, how it will make the new virus, and how it will go to the next generation. So what it is going to prove? Who is entering in the bacterial cell? Phosphorus means DNA is entering in the bacterial cell. So the new virus which is creating inside is produced by which one? DNA. So who is going from one generation to another generation? DNA is going from one generation to another generation. That is finally proved by which one? Hershey and Chase. Now, property of genetic material. Genetic material must have four property. 1, 2, 3, 4. What are these four property? First property, it should be able to make it replica, make it copy. Second property, it must be stable. Third property, it must have a scope of slow change. Fourth property, it must express itself as Mendelian character, like Mendelian character. So out of DNA, RNA and protein, who failed to qualify the first property? Protein. Protein can't be a genetic material because protein is not able to make its replica, protein is not able to make it copy. So who is out? Protein is out. Now how many things are in picture? DNA and RNA. Out of DNA and RNA, who is more stable? DNA is more stable. Who is less stable? RNA is less stable. Why RNA is less stable? Because RNA have ribose sugar. Now question arises, what is the relation with ribose sugar and less stability? So ribose sugar at second hydroxy group in RNA, making RNA, it is more reactive. Second hydroxy group have, second position of the ribose sugar have free hydroxy group, and that is a reactive hydroxy group. So who is more reactive is less stable. So presence of ribose sugar make RNA less stable and more reactive. Second reason why RNA is less stable, RNA act as catalyst. Catalysts are reactive. Who is reactive is less stable. So second reason of less stability of RNA is catalytic nature. First reason, free OH at second carbon of ribose sugar. Third reason, in RNA uracil is there, in DNA thymine is there. So thymine make DNA more stable in compared to uracil in RNA. Got the idea? So these are the reason RNA is less reactive. How many reasons are there? Presence of OH, free OH group at second carbon. Second reason, catalytic nature. Third reason, presence of uracil instead of which one? Thymine in a DNA. These are the things making RNA less stable and more reactive. DNA is more stable because deoxyribose sugar is there. Thymine is there. DNA is non-catalytic in nature, and even in NCERT wrote in a double standard nature of the DNA, complementary base pair stack over each other. Stacking of base pair on the axis provide the additional stability to which one? DNA. So who is more stable? DNA is more stable. So who qualify better the second character? DNA qualify better the second character. Got the idea? It must be chemically and structurally stable. Who is stable? DNA. Who is not stable like that? RNA. Got the idea? Is it clear in the mind? Now, who have a scope of slow changes? DNA. Who can change faster? RNA. See now, in 2 years, how many variant of coronavirus came? It mutate faster because less stable. Are you getting the idea? So second and third character qualified better by whom? DNA in compared to which one? RNA. Based on that, who is a better genetic material? DNA. But last character, who express better as Mendelian character? Here RNA become a winner. RNA can express better as Mendelian character because DNA, when want to express, means making a protein, DNA to RNA to protein. RNA not require the help of which one? DNA. Got the idea? So who can express better? RNA can express better as Mendelian character. But due to more stability and scope of slow change, DNA is more suitable as a genetic material over which one? RNA. And that is the reason, beside of the world is a RNA world. Means who came on earth first? RNA came. But nowadays in most of the organism, who is the genetic material? DNA is the genetic material. Got the idea? Because of a stability and a scope of slow changes. Why we are telling RNA world is there? Right? So RNA, DNA, I am telling you about RNA world. Why I am telling RNA world? So two reason is there. Reason number one, DNA first converting into RNA and RNA then converted into protein. And DNA to RNA formation require enzyme. Got the idea? RNA to protein formation require enzyme. So if DNA came first, how first protein form? Question arises, na? DNA not able to give the answer of from where first protein came. But RNA act as genetic material also and RNA act as catalyst also. RNA act as genetic material also, RNA act as catalyst also. So RNA act as an enzyme also. So RNA have no need of any enzyme to express. So first enzyme formed by which one? RNA. And ribozyme is making RNA to which one? Protein. Understand? And NCERT wrote other thing, process of metabolism, process of translation, splicing, all evolve around which one? RNA. And that is the reason we are telling the world is a RNA world. Are you getting the idea? Is it clear in the mind? So this is over. Now we are moving ahead. Now we are going to talk about which one? Replication part. Replication of DNA. So if we talk about replication of DNA, it is a semi-conservative semi-discontinuous and bidirectional process. Don't forget this. Semi-conservative, semi-discontinuous and bidirectional process. What is the meaning of semi-conservative? Let me tell you. Suppose that this is a DNA. It is going for replication. So both old strand act as a template strand when new strand is forming. And new strand which form is complementary to which one? Old strand. So in both DNA, look, these are the two DNA produced here. In both DNA, one strand is old, another strand is new. It is called semi-conservative. Conservative means two DNA, one old, one new. It is called conservative. Semi-conservative means in both DNA, one strand is old, another strand is new. This is called which one? Semi-conservative mode of replication. Got the idea? Is it clear? And this semi-conservative mode of replication is proposed by which one? Watson and Crick. Who proposed this semi-conservative mode of replication? Watson and Crick have proposed this semi-conservative mode of replication. And the experimental evidences of the semi-conservative mode of replication given by Michelson and Stahl as well as Taylor. It is given by whom? Michelson and Stahl and Taylor. Are you getting the idea? Michelson and Stahl use heavy isotopes of nitrogen, N15, that is a non-radio leveled isotopes. And Taylor use which one? Radio leveled thymidine. Getting the things or not? So Michelson and Stahl work in E. coli, prokaryote, and Taylor work on fava bean, Vicia fava, that is eukaryote. So how Michelson and Stahl prove DNA replication is semi-conservative? Let me tell you. How Michelson and Stahl proved experimentally DNA replication is semi-conservative. Listen it. Listen it carefully. So give your attention. This is the DNA. Red color is showing that both the strand is N15, heavy. Red color strand, red color, both are red color. So both the strand have N15, N15. That is called heavy DNA. Heavy DNA. Both strand are N15. Now they are just allowing then the DNA to replicate. It is E. coli bacteria. This is present in E. coli. In 20 minutes, one E. coli converted into 2 E. coli. Then they remove the DNA from both. So when semi-conservative replication occur, one new strand came. So see, in both DNA, one green strand came. Now it is got the medium density. I am telling semi-conservative. Now after another 20 minutes, means after 40 minutes, 2 bacteria converted into 4 bacteria. Each bacteria have 1 DNA. 4 bacteria have how many DNA? 4 DNA. So when semi-conservative replication occur, what you will find? When replication occur, you just see here, 2 N14 DNA will form, 2 N15 N14 DNA will form, 2 hybrid will form. Are you getting the idea? If you are not understanding, you understand in that way. Here I am taking N15. After one generation, 15 will separate. New will come which one? 14. 2 DNA came. Now next generation, every strand will separate. So this 15 will separate. new came 14. These 14 will new also 14. These 14 new also came 14, and these 14 and new old is which one? 15. So in every generation, how many old will go? 2 will go. Now all new strand will come. So what we are finding? This is medium density, this is medium density, and this is light density, N14, N14. So how many different density is there? 2 different density present in this DNA. Here how many different density are there? Same density are present in both DNA. So when he performed the density gradient centrifugation by taking cesium chloride, he find after one generation, he are getting what? Only one band of DNA. Only one band of DNA, what is the meaning of it? What is the meaning of it, only one band of the DNA? Means they both DNA have same density. Here also same density is coming now. Medium, both are medium. After second generation, he get 2 band of the DNA. What 2 band of the DNA showing? DNA here have 2 different density. So 2 different density came only when DNA replication occur. Which one? Semi-conservative. Got the idea? Understand? Clear? Over? So in such a way, they told DNA replication is semi-conservative. Now from here a question is going to ask. I am asking a question because it is important. It is not important. Numerical questions is important from here. So I am asking a numerical question. Then I will move ahead. So if we grow 40 bacteria containing heavy DNA into a culture medium of N14H4Cl, means where source of nitrogen is N14, then after 60 minutes, how many bacteria contain light density DNA? Light density DNA means 14-14. Heavy means 15-15. Medium means 14-14. Solve this question. So I am solving it. Look here. Give your attention. I am taking a DNA here who is N15 both. Heavy DNA I have taken. I am taking one. Later on I will think about 20. Later on I will talk about which one? 40. 40 DNA, 40 bacteria I am taking. Now I am taking about one bacteria. So in 60 minutes, one bacteria converted into how many bacteria? 20 minutes, one bacteria to 2 bacteria. 40 minutes, 2 bacteria to 4 bacteria. 60 minutes, 4 bacteria to 8 bacteria. 8 bacteria we are getting after which one? 60 minutes. How to handle it? So if you want to go in detail, you go for after one generation, 15, 14. It will also help you, 15, 14. So after first generation, both DNA become hybrid, same density. Now second generation, I am making some speed, 4 strand become 8 strand, 2, 3, 4, 5, 6, 7, 8. First and last is 15. Remaining all will be what? 14. Remaining all will be what? 14. Now it is after second generation, 20 minutes, 40 minutes after, now 60 minutes. So after 60 minutes, 4 DNA converted into how many DNA? 8 DNA. 1, 2, 3, 4, 5, 6, 7, 8. Here 15, here 15. Here 14, here 14. These all are what? 14. Agree or not? First and last stand will be 15, remaining all. So 6 by 8 bacteria have light stand. Agree or not? Light DNA. 6 by 8 bacteria have light DNA. Agree? Now question is what? Question is, I have taken here how many? 40 bacteria. I have taken here how many? 40 bacteria. Are you getting the idea or not? I have taken here 40 bacteria. So 40 bacteria, after 20 minutes 80, after another 20 minutes 160, after another 20 minutes 20, 40, 60, how much? 320. So total 320 bacteria I have. So 6 by 8 are light stand. So what we will write? 320 into 6 by 8. 8 4 ja, 6 4 ja. So total how many bacteria have? 240 bacteria have which DNA? Light density DNA. How many bacteria contain light density DNA? So answer is how much? 240. Everybody understood it or not? Everyone understood it or not? Got the idea? Is it? It is a very good questions from which one? Michelson and Stahl experiment. So is it decoded? Now we are moving ahead. Now we are moving ahead. Give your attention. So experimental verification is over. Now we are going to talk about machinery of DNA replication. So some data is there, you please don't forget. Some data glitch is there, but don't worry, deal like NCERT. NCERT wrote rate of polymerization by DNA polymerase is 2000 base pair per second. Got the idea? DNA polymerase is an enzyme which have a high degree of accuracy, means chances of mistake is very minimal. Got the idea? During DNA replication, deoxyribonucleoside triphosphate serve as dual purpose. What dual purpose? It is acting as substrate also, and it provide energy for polymerization also. Got the idea? It provide energy for polymerization also. Let me tell you about enzyme involved in DNA replication. So very first enzyme we use is helicase. I am telling in NCERT reference, in revision and even in marathon, we are not going to talk about too much detail of the enzyme. So what I am telling, enzyme, helicase. Helicase is the enzyme who break the hydrogen bond. And when breaking the hydrogen bond, what happens? Formation of replication fork occur. So who is the enzyme break the H bond, denaturing the DNA? Helicase. Primase is the enzyme. It initiate DNA replication by making RNA primer. Primer is a RNA primer is a nucleotide of which one? RNA. It is making RNA primer, not DNA primer. Now DNA polymerase is the enzyme who perform polymerization. It is main DNA polymerizing enzyme. And later on, what is there? Ligase. They join the two fragments of the DNA. Are you getting the idea? So just come on the reference of NCERT only. So let's come on the diagram of the DNA replication. Have a look here. See through the diagram. So if we talk about DNA replication, it is semi-discontinuous. One strand making continuously, another strand making discontinuously. What is the reason behind it? Because DNA polymerization always occur in 5' to 3' direction. I am giving you a tricks. Either replication or transcription or translation, they always occur in 5' to 3' direction. Got the idea? So suppose that here it is 3', new strand is forming 5' to 3'. But here if it is 5', if we go for application in this direction, it becomes which one? 3' to 5'. And that is the reason this is the replication fork. One strand making toward the fork, who is 5 prime to 3 prime forming toward the fork, and who is on the discontinuous strand who is forming, it is forming which one? Away from the fork. Right. So it have a 5 prime to 3 prime toward the fork, it have a 5 prime to 3 prime away the fork. Are you getting the idea or not? So the strand of the DNA who is forming continuously is called which one? Leading strand. The strand of the DNA who is forming discontinuously is called which one? Lagging strand. Both the strand forming in which direction? 5 to 3 prime direction only. One strand making toward the fork, another against the fork. So leading strand growing toward the fork and lagging strand growing opposite to the fork, against the fork. It is toward the fork, it is up against the fork. So leading strand is the discontinuous fragment of the DNA first observed by Okazaki, and hence it is called which one? Okazaki fragment. It is discovered by a Japanese scientist. So discontinuous strand is Okazaki fragment. It is called Okazaki fragment in the honor of the discoverer Okazaki. Got the idea? Right. So template strand for leading strand is 3 prime to 5 prime. Template strand for lagging strand is which one? 5 prime to 3 prime. And DNA replication is what? Basically semi-conservative, semi-discontinuous, one strand continuous, another strand discontinuous, and bidirectional. It is not bidirectional. It is bidirectional means from ori site it occurs in both the directions. So DNA replication is what? Basically bidirectional. Now we are going to talk about which one? Transcription. And what NCERT wrote, and it is also a very important line. You please don't forget it. I am underlining that line. That is a highly important line. Don't forget it. And what is the line? If a failure of cell division after DNA replication result into which one? Polyploidy. It is a chromosomal anomaly. Right? If DNA replication occur and cell divide to fail to divide, who will occur? Polyploidy occur. Got the idea? Now we are going to talk about transcription. What happened in transcription? Formation of RNA. In transcription, what happened? Formation of RNA occur from DNA. Got the idea? RNA formation occur from DNA. So DNA is a double stranded structure, RNA is a single stranded structure. So during transcription, only one strand of the DNA act as a template strand. Only one strand of the DNA act as a template strand because RNA is a single strand. During replication, both strands act as a template strand, but during transcription, only one strand acting as a template strand. Got the idea? NCERT wrote further, what possible problem occur when both strands act as a template strand. So assume this is also a template, it is also a template. So both will make a RNA. Both will make a RNA. So this is different mRNA and this is different mRNA. This will forming X different protein, this is forming Y protein. Unnecessary confusion occur. Same DNA, when protein, X protein, when same DNA, when form X protein, when form Y protein. Got the idea? Another possible problem is what? Both the DNA, RNA strand are complementary and they join and they make which one? mRNA, who is double stranded. If double stranded mRNA form, who will not occur? Protein synthesis not occur, because during protein synthesis mRNA pair with tRNA. If it is already pair, how it will pair with the tRNA? Understanding the idea? And that is the reason out of the two strand, only one strand acting as a template strand to avoid unnecessary confusion. Otherwise, if both act as a template strand, two RNA will form. So protein synthesis become a too confusing process. Now we are going to talk about next paragraph, that is which one? Transcriptional unit. So a transcriptional unit consists of three components. Please don't forget. How many components are present in a transcriptional unit? Three components: a promoter, a structural gene, and a terminator. These are the components of a transcriptional unit. Now I am showing you the diagram of a transcriptional unit. Now I am showing you a diagram from a transcriptional unit. and we will deal some numerical question from here. Every year one question came. So be careful here. Here I am going to deal very important things. So see, this is a transcriptional unit. It is a promoter, it is a terminator, and this is a structural gene. Got the idea? Now, coding a strand used as a reference. Coding a strand are that strand always who are from 5' to 3'. So strand of the DNA who is 5' to 3' is called coding strand, and strand of the DNA who are 3' to 5' is called template strand. Who act as a reference? Coding strand act as a reference. So if it is coding strand, so it is what? 5' to 3'. So promoter is always present toward which one? 5' of the coding strand. So this is promoter, and here it is terminator. So promoter is present in upstream of the structural gene, and terminator is present in downstream of the structural gene. Terminator is present in which one? Downstream of the structural gene. Even promoter also used as a reference. Suppose that here it is promoter, so this strand will be 5' to 3', and this strand will be 3' to 5'. I am drawing a diagram, you will understand it better. Give me a moment. It is important to understand that the reason I am telling it. So wherever important is there, we will not do the hurry. Look here, give your attention. Here it is the DNA, transcriptional unit. Here I am telling promoter, here I am telling terminator, now I am asking who is here strand A, here strand B. Who is 5' to 3'? A strand A is 5' to 3'. Why? Because near the promoter who is there 5', and here near the promoter 3'. So we will read it 5' to 3', and we will read it 3' to 5'. So 5' to 3' when we are reading, it is which strand? Coding strand. And here it is which strand? Template strand. Now we are changing it. What change I am making? Next time I am writing everything like this, but I am changing here. I am doing terminator, here I am doing promoter. Now here it is A strand, here it is B strand. So here B strand is what? 5', it is this. So 5' to 3', it is called coding strand. And it is called which strand? Template strand. Template strand is always 3' to 5'. Coding strand is always 5' to 3'. Are you getting the idea or not? So whenever I am changing the promoter, who change? Coding strand change, na? Earlier upon were the coding strand. Now who is the coding strand? Lower one is the coding strand. Is it clear? So both are referential to each other. Coding strand and promoter both use as a reference. Getting the things or not? So when we are changing the, now I am writing a very important line. Very, very important line. And what is very, very important line here? Let me tell you. What is very important line here? Have a look. Oh. RNA is complementary to template strand and like coding strand. Every year one question came from here. What I wrote? RNA is complementary to template strand and RNA is like coding strand. I am writing here. Only difference is what? Only uracil came in place of which one? Thymine. I am writing here. Suppose that here I am writing A, T, C, G, G, C, T, A, G, C, G. It is in DNA. So here B is which strand? B is which strand? Template strand. Template strand, and A is which strand? Coding strand. Coding strand is also called sense strand. And template strand is also called anti-sense strand. It is also called which one? Anti-sense strand. Are you getting the idea? Now give your attention. What will be the mRNA? mRNA always form in 5' to 3' direction. Transcription always occur. I told you, replication, transcription, translation, everyone always occur in 5' to 3'. So here T is there. T complementary is A. G complementary is C. C complementary is G. A complementary is T, but T will not come, U will come. G in thymine not present here, na? G here complementary C will come. So this is the RNA. Assume this is mRNA. So what I told, mRNA is complementary to which strand? Template strand. And mRNA is like whom? Coding strand. Why I wrote like? Why I wrote like here? Look here. This is A, this is A, this is C, this is C, this is G, this is G. Thymine is not present here, so U came. Cytosine, cytosine. It is 5' to 3'. It is also 5' to 3'. So mRNA is like the coding strand, and mRNA is complementary to template strand. And based on that, I have some bits on it. You try to solve it. I'm not writing the bits. I have a bits of the previous year question. So I will just specifically focus on that bits. Probably here is the questions there. Let me open. I will not focus on any other question other than Here you just try to, it is 2019 Neet question. Oh, no, it is not related to this. Wait. Another question I'm bringing. What happening? Why it is not scrolling? Wait a minute. I want to show you these questions basically. Where is that file? Finder. It's supposed to make PDF. I not made it PDF here. This is 2018 Neet question. We are just going to deal it. Here you solve it. Question is, this is the sequence from the coding strand. You just mark it. It is a sequence of coding strand of a gene. What will be the corresponding sequence of the transcribed mRNA? It is Neet 2018 question. What the idea? So what is there? We know coding strand. mRNA is like the coding strand, only who differences will come? What differences will come? What differences will come in place of thymine? Who will come? Uracil. So adenine AGG remains same. So which option have A, G, G same? A, G, G same. Got the idea? In place of T, who came? U came. Now A will A, in place of T, U will come. Now C, G, C, A remain same, and in place of T, who will come? U. So answer is third. Got the idea? Understand? Now one more question we are going to see from here. Have a look. See this question. It is also a P by Q. Here I not wrote here. See this question. What is the question? Which one of the following is the sequence of corresponding coding strand? They are asking coding strand sequence. And they have given which one? Sequence on mRNA. So who is same? mRNA and coding strand are same. That time they ask mRNA, this time they are asking coding strand. Both are same. So in mRNA, who is there? Uracil is there. In coding strand, in place of uracil, who will come? Thymine will come. So here, very first A is there. So A will come. A will come. So A is not present in first option. A is not present in fourth option. Two option eliminated. A is present in how many option? Two option, 2 and 3. So A will come. Second, who will come? Second, who will come? Second, who will come? U. So T will come. AT, both have. Third, who will come? CGA come. CGA came. CGA came. Are you getting the idea? Later on, U came. U place, who will come? T came. It is going same. Got the idea? Later on, who will come? CGA will come. CGA will come. Later on, T came. It is coming like that. Now CGA will come. CGA will come. Here also CGA is there. T, it is going all together. Na? Got the idea? You got the problem, na? How to handle it? CGA, CGA. Are you getting the things? So what we will find? We will find like this. It is simply a wastage of time, so I am not going for everyone. Getting the things now. So how to handle? Simply what is in mRNA, same came in coding strand. What the only differences will come? Only differences in place of uracil, thymine will come. Got the idea? Right? So in searching it, waste our lot of time. We are only running out of the time. So wait a minute. Running out of the time now. So wait a minute. So have a look. Any more question is there based on that? Because in every year one question coming like this. Every year you will find one question of such type of coming. DNA given, find the RNA, got the idea, it is of another chapters. Okay, so leave it now. Okay, done. I hope you understand how to handle such type of question. Is it clear? Right. Now we are moving ahead. Give your attention. So this is over. Now NCERT wrote one line, you also must know that some regulatory sequence may present before promoter and after promoter. Some regulatory sequence either present before a promoter or present after the promoter. This is also written in this paragraph. It is over. Now we are going to move on the next. Transcriptional unit and the gene. Let me speak on that. Transcriptional unit and the gene. What NCERT wrote about it? NCERT wrote that gene is a unit of inheritance, we know better. But it is also very clear that gene is present on which one? DNA. But it is difficult to define gene as a segment of DNA. Got the idea? You cannot say about, sir, look, this segment of the DNA is a gene. NCERT wrote it is difficult to define gene as a segment of which one? DNA. NCERT further wrote that the DNA sequence who are coding for, the DNA sequence who is coding for tRNA or mRNA also define that gene. Why NCERT wrote like this? Because earlier people believe that gene is only that segment of DNA who is making protein. Means earlier people believe that gene are that segment of DNA who is making only mRNA. But NCERT author wrote here what? The segment of DNA who is coding for mRNA, sorry, tRNA and rRNA is also defined as gene. And further then NCERT told, introduce a new term, cistron. Earlier, we can say now, modern name of the earlier old classical gene is what? Cistron. Earlier we think gene are that segment of DNA who are making a polypeptide. Now we are thinking what? Cistron are that segment of DNA who is making which one? Polypeptide. And eukaryote are usually monocistronic and prokaryote are usually polycistronic. What is polycistronic? Polycistronic means one cistron have information for more than one polypeptide. It is a character of prokaryote. What is monocistronic? One cistron have information for one polypeptide. Further NCERT wrote, a split gene arrangement in eukaryote. A split gene is what? Exon, intron means exon is coding sequences, intron are non-coding sequences. So exon is interrupted by which one? Intron. So NCERT wrote a split gene arrangement further complicate the defining the gene as a segment of DNA. Exon are that part of DNA whose information is present in process mRNA. Intron are that part of DNA whose information is not present in process messenger RNA. Are you getting the idea? So this is about what? Exon and intron NCERT wrote. Later on here student confuse a lot. Got the idea? Where student confuse a lot? Let me tell you, I am just underlining this line and I am telling you, have a look here. Inheritance of a character is also affected by promoter and regulatory sequence of a structural gene. Hence, sometimes the regulatory sequence are loosely defined as regulatory gene, even though these sequence do not code for any RNA and protein. Now, a student came to us and asked, sir, regulator gene making a protein, na? NCERT wrote here, not making RNA and protein. NCERT want to say regulatory sequence doesn't mean only regulator gene. Promoter also coming under regulatory sequence. Operator coming also under which one? Regulatory sequence. Regulator gene also coming under regulatory sequence. So NCERT author simply wrote here in the flow. You just think like promoter, operator, not making any protein. But beside of that, if promoter is not there, who will not occur? Protein synthesis will not occur. So NCERT author want to say all that segment of a DNA who are either directly or indirectly involved in protein synthesis is called gene. And the segment directly involved in the protein synthesis is called what? Cistron. So the segment directly have information for polypeptide formation is called cistron. But all that segments who is directly or indirectly involved in protein synthesis is a what? Gene. Got the idea? Done? Clear? Now we are going to talk about which one? Type of RNA and the process of transcription. If we talk about the type of RNA, you better know three type of RNA here. Ribosomal RNA. Ribosomal RNA is most stable and most abundant RNA. It is a component of ribosome. Right? Now a messenger RNA is least stable RNA. It have information of DNA for protein synthesis. And transfer RNA is second most abundant and most stable RNA. Got the idea? And it transfers the amino acid at the site of protein synthesis. It acts as an adapter molecule. It is the smallest RNA as well and also called soluble RNA. Now we are going to talk about in prokaryote, all three RNA produced by only one enzyme. That is what? DNA dependent RNA polymerase. All three enzymes in prokaryote formed by only one type of RNA polymerase. Don't forget this line. But in eukaryote, class stop in the app. One student is writing. Is it for all? It may be required to restart, it may be auto stop it occur. It is coming in app class, is coming for all or not coming? Not coming. Okay, let me call the operator. Give me a minute. It may be after a longer duration, they stop automatically. Wait a minute. हाँ, come once Ravi Garu. हाँ. Wait a minute. In app, it is not coming. So once it's restarted.
Oh, okay. 5 hours.
So after a particular time, it stop na.
5 hours also starts.
Came.
Students are joining. 5 minutes. Students are joining.
2 minutes.
Five hours also video Botany, sir.
After 5 hours? No, it is online. Okay, we will talk later. Audible? Is it audible now? It is coming. Okay, ji. So we are going to talk about what I told in prokaryote, all 3 type of RNA is produced by a single enzyme, that is DNA dependent RNA polymerase. But in eukaryote, there are 3 different enzymes there, and this is also a potential question. RNA polymerase produce DNA too. I am writing also. It is a potential question. Easy, I know every one of you. So DNA to rRNA. It is performed by which one? RNA polymerase 1. RNA polymerase 1. Which rRNA? 18S, 28S, and 5.8S. Now DNA to hnRNA. In eukaryote, directly mRNA not produced. Heterogeneous nuclear RNA. It is a precursor of mRNA. It is by 2. And DNA to tRNA, snRNA, and 5S rRNA produced by RNA polymerase 3. Don't forget this data. Now further, what are the steps of transcription? What are the steps of transcription? So transcription occur in 3 steps: initiation, elongation, and termination. And here sometimes student confuse. Let me what happened. During initiation, first what happened? RNA polymerase, along with RNA polymerase, who is binding? Sigma factor is binding. It is initiation factor. Initiation factor. And then they together bind at which one? Promoter. So initiation done by whom? RNA polymerase plus sigma factor. Alone sigma factor cannot initiate. Who have a role also? RNA polymerase have a role also in initiation. So RNA polymerase plus sigma factor, initiation factor, it initiated a transcription. As the transcription started, who detached? Sigma factor detached. And elongation only performed by RNA polymerase. At certain time it is written core enzyme. Got the idea? It is performing the which one? Elongation. Now when the RNA polymerase reach at terminator site, here another factor join with them, rho factor, and it is do the termination. So RNA polymerase have involvement in all 3 processes: initiation also, elongation also, termination also. Right? But initiation RNA polymerase cannot do alone. It do along with the help of which factor? Sigma factor. Elongation RNA polymerase doing alone. But termination it need the support of which one? Rho factor. Understanding the idea? Is it clear in the mind? So this is the 3 steps of the transcription: initiation, elongation, and termination. And complementary to template stand, new mRNA is going to produce during write. Now further NCERT wrote in prokaryote, transcription and translation is a coupled process. Coupled process means occur together. Why it occur together? Because in prokaryote, both transcription and translation occur in cytoplasm itself. Both transcription and translation occur in cytoplasm itself at the same location. Another thing, in prokaryote, mRNA produced directly, means no processing required like eukaryotic mRNA. Understanding the idea? And that is the reason it is a coupled process, occur side by side. Even one side who is going on, transcription going on, another side who started, translation started. That's the reason it is called coupled process, it occurred together. Got the idea? But in the case of eukaryote, transcription and translation is not a coupled process. Reason is 2. Reason number 1, transcription occur in which one? Nucleus in eukaryote, translation occur in cytoplasm. Further in eukaryote, directly mRNA not producing, DNA forming hnRNA in nucleus, and further hnRNA undergoing in processing. What happened in processing? What happened in processing? In processing, 3 events occur: capping, splicing, and telling. Capping, splicing, and telling. So hnRNA undergoing in capping. During capping, what happened? 7-methyl guanosine triphosphate join at 5' end. During a splicing, what happened? Removal of intron occur. It occurs with the help of enzyme complex that is called a spliceosome complex. And in telling what happened, polyadenylation occur. 200 to 300 polyadenine join at which one? Template independent manner. And then a mature process mRNA is forming. So in eukaryote, processing required, and these processing require, it is requiring time, and that is the reason transcription and translation is not a coupled process in which one? Eukaryote. Are you getting the idea? Is it clear in the mind? Done, over. Now we are going to talk about genetic code. Genetic code, I am telling the salient character. Genetic code are triplet, first thing. Genetic code or triplet is told by George Gamow. What the idea? Another scientist name you don't forget, Hargobind Khurana. He developed the chemical method and synthesized RNA polymer artificially. He make the two type of RNA polymer. One is homopolymer, another is copolymer. Homopolymer means only one type of nucleotide is there. Heteropolymer means more than one type of nucleotide is there. Another scientist you don't forget, that is Severo-Ochoa. He developed an enzyme that is called Severo-Ochoa enzyme. And what this enzyme is doing, it performing the polymerization of RNA in template independent manner in vitro. And third is which one? Marshall Nirenberg. Marshall Nirenberg developed the cell-free medium of the protein synthesis. Outside the cell, how protein synthesis occurs, that technique is developed by which one? Marshall Nirenberg. Got the idea? Is it clear? So these are the some of the discovery finally help us to decipher the character and properties of the which one? Genetic code, and told the genetic code are triplet. If it is possible for you people, please take care about the which one? Genetic code. Once, twice, try to read it, try to fix it, because sometimes question going to ask from genetic code also. They will give the which one? Sequence of code, and they will ask which amino acid coded by there. This is quite possible, so please take care of it. Okay, now we are going to talk about property of genetic code. So if we talk about property of the genetic code are triplet, means 3 nitrogenous base making 1 code. Second property are genetic code are contiguous. Contiguous means no space, no comma, no bracket, no sign of punctuation present between the two code. Third character is genetic code are non-overlapping. Non-overlapping means suppose that I am writing a code AUG and I am writing GUC. So it is correct method of genetic code. You are not going to suppose to say, sir, look, one code, another code. It is wrong. These two are overlapping between the two code. It is correct method. It is incorrect method. Now next is genetic code are nearly universal, with few exception of some mitochondrial gene and protozoan gene. Most of the code have same meaning in every species. What is the meaning of universal? GUG or GUC, an example, coding for valine in human being also, plant also, bacteria also, reptile also. So a code have every meaning in every same meaning in every species. It is called universality. Now genetic code are non-ambiguous. Non-ambiguous means what? Non-ambiguous means what? Non-confusing. A genetic code have always one meaning. One word, one meaning. Is what? Non-ambiguous. One word have same meaning in every species, universal. Like AUG code for methionine, not any other amino acid. That is called non-ambiguous. And genetic code are degenerate. Means more than one code are responsible for one amino acid, like GUG, GUC, GUU, GUA, all code for which one? Valid. Now NCERT asks a beautiful question here. Look here. Wait. What happening? NCERT is saying this is the sequence of nitrogenous base on mRNA. Can you tell about the sequence of amino acid? Yes. AUG only code for methionine. UUU code for? Tell me. Fast. Who are at least online, tell me. UUU code for phenylalanine. UUC code for phenylalanine. Methionine. It is phenylalanine. It is phenylalanine. It is phenylalanine. It is phenylalanine. So we can easily say sequence of amino acid if we know the sequence of mRNA. It is possible due to which property of genetic code? Non-ambiguity. It is possible due to non-ambiguous nature of the genetic code. Are you getting the idea or not? But can you say mRNA sequence from this amino acid sequence? No, we can't say, because phenylalanine is there. Here either UUU came or here either UUC came. How can we guarantee? So we cannot say mRNA from amino acid. It is due to which property of the genetic code? Degeneracy. It is due to degeneracy of the genetic code. Did you got the idea? So this is about the genetic code. Now we are going to talk about mutation and genetic code. Just have a look. So I am telling every mutation here only. So if we talk about mutation, gene mutation, if we talk, gene mutation or point mutation is of two types: substitution mutation and another is frame shift mutation. In substitution mutation, what we are finding? Purine is sorry, wait a minute. I am in hurry somehow. Wait a minute. Wait. All will go. Okay. Let me take this another slide. There, if anything you wrote wrong, it becomes difficult to erase. So I am telling gene mutation. 2 minutes, 3 minutes I will take, I will finish it. It is of two types. One is substitution. Another is frame shift. If we talk about substitution, it is two types. Transition. Another is transversion. Transition, what is there? Purine is replaced by purine and pyrimidine by pyrimidine. Transversion, purine replaced by pyrimidine. Simply I am writing by pyrimidine. Replace I am not writing, and vice versa. It is called transversion. Got the idea? This is substitution mutation. Example of transversion: sickle cell anemia. It is very important. Sickle cell anemia is a beautiful example of transversion. What happened in sickle cell anemia? In mRNA, GAG replaced by which one? GUG at 6th position. At 6th position. Got the idea? GAG code for which one? Glutamic acid. GUG code for which one? Valine. How I am telling? It is mRNA, na? 5' to 3'. It is 5' to 3'. In DNA, who is there? 5', GAG, 3', CTC, 3' to 5'. Here who is there? GTG. Here it is one CSE. It is DNA. Mutation occur here. You just look here, mutation occur. Here mutation occur. A replaced by T. Adenine is a purine. Thymine is a pyrimidine. Did you got the idea? A replaced by T. Purine replaced by pyrimidine. Due to that reason in mRNA, when no mutation occur, before here mutation occur. Before mutation, it is giving GAG coding for glutamic acid. After mutation, it is making GUG, it is coding for valine. And it occur in which one? Beta chain of hemoglobin, and at which position? 6th position. Right? That I also finish here only. You got the idea? Understand? Clear? Over. So this is the example of which one? Transversion. Now we are going to talk about frame shift mutation. Frame shift mutation is of two types. One is what? Addition. Another is what? Deletion. Got the idea? Addition and deletion. So very important, addition or deletion of one or two nucleotide cause frame shift. Addition or deletion of three or its multiple nucleotide not cause frame shift. Got the idea? And NCERT have given a beautiful example for that. What beautiful example is there? We are going to discuss. So NCERT wrote, Ram has red cap. When we are adding B before R, reading sequence change, code change. When we are adding BI, 2 nucleotide, reading sequence change. But when we are adding 3 nitrogenous base, red remain rem, cap remain cap. So addition or deletion of 1 or 2 nitrogenous base causing the reading frame change, 3 or its multiple nitrogenous base not change. Same happen in the deletion side. You got the idea? Now we are going to talk about tRNA as adapter molecule. Got the idea? tRNA as adapter molecule. See, what is the problem? NCERT wrote, from the very beginning of the proposition of the code, Francis Crick know that, got the idea? There has a mechanism to read the code. Problem is what? Amino acid cannot read the code. Amino acid and which one? Genetic code are not complementary. Genetic code and amino acid are not complementary. Now, how amino acid bind with a particular code? Got the idea? It is become possible due to which one? Transfer RNA. How? tRNA is responsible for identification of a particular amino acid. And especially D arm of the tRNA have this power. So D arm of the tRNA identify, if we talk about the tRNA brief structure, wait a minute. The 2D structure of tRNA is like a clover leaf. I am making this structure. It is a clover leaf like a structure of tRNA and proposed by Hawley. It is D arm. It is anticodon arm. It is T size C arm. It is variable arm. And it is 5 prime. It is 3 prime. It is always terminate with CC. It will always start with G. So this is a 2D structure of tRNA. It is like a clover leaf model. 3D structure is like inverted leaf. Sorry, 3D structure is inverted L-shaped structure. Now this D arm recognize a specific amino acid. A particular tRNA responsible for a specific amino acid, and these DR recognize a special amino acid by recognizing enzyme attached with that amino acid. And after identifying these amino acid attached at which one? 3'. 3' is amino acid attachment site. It is amino acid attachment site. And these anticodon arm is complementary to a codon. Now suppose that this is a tRNA, try to understand, who have a anticodon arm UAC. The tRNA who have a anticodon arm UAC, they bring a special amino acid whose name is methionine. Now these tRNA came and bind at mRNA with codon. So UAC pair with which one? AUG. And it is bringing which amino acid? Well, which one? Methionine. So methionine came in front of which one? AUG. Neither AUG no methionine nor methionine no AUG. So who acting as a mediator between the two? Who is knowing both the people? tRNA. tRNA acting as an adapter. So they are bringing methionine and placing them in front of which one? AUG. Likewise, are you getting the idea? So what the job tRNA is doing? tRNA one hand recognizing amino acid, another hand they are recognizing mRNA, and they are arranging a specific amino acid in front of a which one? A specific codon of the mRNA. Are you getting the idea? Right? Is it clear in the mind? So how tRNA doing this job? You are getting the things, understanding the idea? Right? So tRNA is the smallest RNA, so it is called which one? The soluble RNA also. Got the idea? Isn't it? Anticodon loop of the tRNA is complementary to which one? Codon of the which one? mRNA. Amino acid bind at which prime of the tRNA? 3' of the tRNA. Understanding the idea? Who recognize the amino acid? DR recognize the amino acid. Who pair with codon? Anticodon arm pair with the codon. It is called which arm? Anticodon arm. It is written already. T psi C join at ribosome. Means its job is to bring the amino acid at the site of protein synthesis. So this is about the tRNA. Now we are going to talk about protein synthesis, translation. So in translation, what happened? Let me tell you. What you must not forget from the translation is, what must not forget from here is, so translation is a process of polymerization where so many amino acids join together by peptide bond. And sequence of amino acid in a polypeptide is decided by which one? Sequence of nitrogenous base in the genetic code, mRNA. mRNA deciding the sequence. Got the idea? Because if AUG is here, methionine will come, UUU is here, phenylalanine will come, GUG is here, valine will come. So which code is there? That based on that, who will come? mRNA will come. So now further NCERT wrote that in a polypeptide chain, 2 amino acids join together by which one? Peptide bond. And these peptide bond formation require energy. Question arises, from where this energy is coming? So NCERT wrote that therefore in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, right? Commonly called charging of tRNA or amino acylation of tRNA. So when with tRNA who join, when I was teaching first time I told in detail, here amino acid is joining. So during charging process or amino acylation process, who is requiring? ATP is requiring. And when this tRNA and amino acid detach, energy release, and these energy used in the formation of peptide bond. Got the idea? Later on NCERT wrote that this peptide bond formation is catalyzed by an enzyme, that is what one? Ribozyme. Ribozyme. And these ribozyme is chemically RNA, ribosomal RNA. Who is acting as a ribosome? 23S ribosomal RNA act as ribozyme, and this ribozyme is which enzyme? Peptidyl transferase. So let me say about it. It is important to know. Peptide bond formation is mediated by a enzyme peptidyl transferase. What is the name of enzyme making peptide bond? Peptidyl transferase. Chemical nature of the peptidyl transferase is ribosomal RNA. Which ribosomal RNA? 23S ribosomal RNA in prokaryote. Because here RNA acting as an enzyme, so it is a what? Ribozyme. Are you getting the idea? Understanding the idea? Later on what NCERT is writing about the protein synthesis. Let me move ahead. I am just moving NCERT referential. So just move ahead. Charging and all are done now. Now give your attention. Energy and everything we just found. Now just see here. Now further NCERT writing. What happening? These ribosome are there. So these larger subunit have two sites. One is P site, another is A site. What the idea? P site is peptidyl site and A site is amino acid site. So look, what happening? In very beginning, what happening? In very beginning of the protein synthesis, what's happened? This is mRNA. Along with mRNA, smaller subunit of ribosome came and attached. These are the smaller subunit of ribosome. These are initiation codon, another codon like this. Got the idea? Later on, who came? Initiator tRNA complex came. Methionine came. Then larger subunit joined. Here it is P site. Here it is A site. Now P site is occupied. A site is free. Initiation is over. Now during elongation, what happened? Another charged tRNA came. Another charged tRNA came and joined at which site? A site. As both P and A site will occupy it, both join together by which one? Peptide one. From where energy will come? Energy will come when this detach from here. Now ribosome move by one triplet toward 3'. Now they are covering this triplet. Now when amino acid move further, when ribosome move further toward 3', the tRNA at P site fall in the cytoplasm. Now every time A site become free and new charge tRNA came here and it is joining, further peptide bond forming, it is breaking, it is joining, further it is moving toward the 3'. This is what basically elongation is going on, and this process will continue as long as a stop codon will not come. And when a stop codon came, a release factor going to dismantle every process and protein synthesis stop. Got the idea? Right? And then further NCERT is saying one thing more. Here I am just drawing a diagram. That is translational unit they are telling. So this is the cap. This is translational unit. This is tail. So between cap and tail, both sides is there, UTRS is there, and translational region is there. Here start codon, here stop codon. So start codon and stop codon is flanking the translational unit. Translational unit. One side start, another side stop codon. Between start codon and cap, UTRS is there, and between stop and codon and tail, UTRS is there. These UTRS required for efficient translation. Efficient translation. Got the idea? Now we are moving toward which one? Regulation of gene expression. Regulation of gene expression. Have a look. So regulation of gene expression, NCERT wrote, in prokaryote, it occurs only at transcriptional level. But in eukaryote, these regulation of gene expression occur at 4 different levels. Maybe transcriptional level, maybe processing level during regulation of splicing it to, transport level from nucleus to cytoplasm of mRNA, and translational level. So who have a better control over his gene? Eukaryote have a better control over a gene. Regulation always occur for a luxury gene. There are 2 types of gene present in our body. One is housekeeping gene, that is called constitutive gene. Another is luxury gene. Housekeeping gene working all the time, luxury gene working on demand. So who need regulation? Luxury gene require regulation. Housekeeping gene never require regulation. Constitutive gene not require regulation. Regulation means what? When a gene form a protein, when a gene not form a protein is called regulation. Got the idea? Understand? Clear in the mind or not? Now I am telling regulation of gene expression how regulation of gene expression occur? This is elucidated by Jacob and Monod. Jacob and Monod have given the concept of switch on and switch off. They give the concept of switch on and switch off. What Jacob and Monod told, I am just telling you. They talk about the regulation of gene expression in which one? E. coli, and regulation of lactose metabolism. So they told which one? Lac operon. Lac operon. What Jacob and Monod told in the lac operon, I am telling you. Have a look here. So here two type of gene is there. Just try to understand. One is I gene. It is called regulator gene. Another is here Z, Y and A gene. It is which gene? A structural gene. So structural gene is a luxury gene. I gene is a constitutive gene. They are synthesizing protein all the time. Constitutive gene work 24/7 into life. So constitutive gene control the regulation of a structural gene. How? Let me tell you. These constitutive gene going for transcription. Why? How it is going for transcription? First RNA polymerase will bind at promoter. Then RNA polymerase move here. And then what happen? Transcription occur. Here mRNA produce. Now after mRNA production, translation occur and protein forming. This is repressor protein and this is active repressor protein. Active repressor protein. Now what happened? When active repressor protein, it is also called as which one? Accessory protein. When you read the initial line of NCERT, it is written like this. So whenever these active repressor protein binding with operator, RNA polymerase, it is going to partially cover the promoter also, and RNA polymerase for which gene? A structural gene not able to bind to promoter. Who is not accessible? Promoter size is not accessible for RNA polymerase of the structural gene. Whenever who is binding? Repressor protein is binding at operator. And when promoter is not accessible, what happens? These structural gene not transcribe, not translate, so protein synthesis not occur. So these structural gene is in which mode? Switch off mode. Switch off mode. So who is making them switch off? These regulators gene making as a switch off. These regulators in I acting as an inhibitor here. Acting as an inhibitor here. Understanding the idea? They are acting as an inhibitor here. They are not allowing the structural gene to synthesize any protein. They are not. Why it is not forming protein? Because their RNA polymerase is not able to bind at promoter due to blocking of which part? Operator region. Now, what happened? Whenever lactose present in the environment of cell, who is present in the environment of cell? Lactose is present in the environment of cell. So lactose, when present in the environment of cell, lactose acts as a which one? Inducer. What lactose is doing? Here who is the inducer? Here inducer is lactose or allolactose. So they bind with repressor protein. Who is making the protein? Who is making the protein? Regulator gene. But who is making them inactive? Lactose or allolactose, which is an inducer, it is binding here. Now what happened? When these become inactive, it is not able to bind at operator. Operator is free. So who is clearly visible? Promoter is clearly visible. RNA polymerase bind at promoter size. Transcription occur. Transcription occur. It is forming mRNA. This mRNA is a which kind of mRNA? Polycystonic mRNA. Because it is ZYA3 gene information is here. Now translation occur and Z will form beta galactosidase, Y will form permease, and gene A will form which one? Trans-acetylase. Are you getting the idea? It is forming which one? Trans-acetylase. So beta galactosidase digest lactose or beta galactoside. What is the function it is doing? Permease have role in absorption of lactose. NCERT not wrote about trans-acetylase. I am not also saying here right now. So these all the enzyme have role in lactose metabolism. You just assume for a minute, if lactose is not present in the environment of the cell, is there any job done by beta galactosidase? It digests lactose. It is requiring only when who is there? Lactose is there. Whenever no lactose, no need of the which one? Beta galactosidase. That is the reason how nature works smartly. When lactose came in the cell, then only they are releasing these enzymes, because these enzymes are only associated with when lactose is present. So when lactose is present, this gene gets switched on. This gene get switched on. And when lactose is not there, the gene gets switched off. Otherwise, if lactose is not there and this enzyme is releasing, it is a useless job, na? Understanding the idea or not? Clear in the mind or not? Right? Now further NCERT wrote, okay, they ask a question. If lac operon not working, suppose that this is a bacterial culture medium, here bacteria is there, here lactose is there. For absorption of lactose, who needed? Permease needed. So if permease is not there, how lactose will absorb by the bacterial cell? So NCERT give the answer of this question also. A very low level of lac operon working all the time. And further what happening? Lac operon is a which kind of operon? It is which one? Inducible operon. Inducible operon. Who is inducible operon? Lac operon is an inducible operon and under negative control. You studied till now negative control. Further NCERT wrote, it is under both positive and negative control. How it is under positive control, you will study in the higher class. But at this standard, they are writing it is an inducible operon and it is under which one? Negative control, and it is better to say it is under which one? Both negative and positive control. So this is about the lac operon and this is about the molecular basis of inheritance. Now we are going to talk about which one? Mendelian inheritance, and I am leaving NCERT because it is going to slow my speed. If I not going to stay with the NCERT, we can say cover every line. NCERT using is going to slow the speed. Principle of inheritance. Got the idea? Principle of inheritance we are going to discuss. So look here. We are just going to talk about inheritance of one gene. Inheritance of one gene. What is important from here, you don't forget. So in F2 generation, phenotypic ratio is 3:1. Genotypic ratio is 1:2:1. And in inheritance of one gene, in dominance, F1 resemble with either of the parent, not both, any one parent. What is the idea? If you just go for, we are making a cross of tall versus dwarf, parent Mendel always taken pure line, capital T, here small t, capital T gamete, a small t gamete, F1 will capital T, small t. When we grow, it is tall plant. And when what Mendel did, selfing of F1, capital T, small t, capital T, small t, capital T, small t, capital T, small t. These are the gamete. This is the Punnett square, capital T, capital T, capital T, small t, capital T, small t, small t, small t. So what happening here? What the phenotypic ratio he got? He got it is tall, it is also tall, it is dwarf. So phenotypic ratio is which one? 3:1. Tall is 75%, dwarf is 25%. Genotypic ratio is capital T 1, capital T small t 2, small t 1. So this is the monohybrid cross. Based on monohybrid cross, what told by the Mendel? Got the idea? Based on monohybrid cross, Mendel given Mendel given two law. First, based on monohybrid cross, Mendel proposed two law. One is law of dominance law of dominance, another is law of purity of gamete or segregation of gamete. Got the idea? In law of dominance, Mendel told three things. What Mendel told in law of dominance? Factors is present in which one? Pair. You just see the diagram of the NCRT. Here NCRT line is important. You just see. Give a minute. Give a minute to me. Finder. Principle of inheritance. What happening? So these three things you don't forget. It is very important. So in law of dominance, what Mendel told? First thing Mendel told, what? Character is controlled by discrete unit called factor. Discrete means separate, who not blend. So character are controlled by separate unit called factor. Both factor T or capital T or small t, they never blend, they never mix, they always remain separate. Second thing he told, factors occur in pair. Third thing he told, in a dissimilar pair of factor, one member dominate the other. Suppose that here capital T small t, there it is tall. So who is dominating? Capital T is dominating. Who is suppressing? Small t is suppressing. And NCRT further wrote, the law of dominance is used to explain the expression of only one of the parental character in F1 generation and both in F2 generation in which ratio? 3:1. So expression of only one parental character in F1 generation and both the character in F2 generation is explained by which one? Mendel law of dominant. Now law of segregation of gamete. These are the second law Mendel proposed, also based on which one? Monohybrid cross. What Mendel told in law of segregation of gamete? The NCRT wrote, the law is based on the fact that allele do not show any blending. Allele always present as discrete unit. So if not, and that both the character are recovered as such in F2 generation. You just look, parent is tall and dwarf. It is parent generation. If we talk about F1, it is tall. If we are making selfing of tall, in F1 we are getting tall versus dwarf. In which one? 3:1 ratio. So both are expressing both character are recovered in which one? F2 generation. This is possible due to which one? Law of segregation of gamete. Dwarf is not visible in F1, but it is visible in F2 generation. Are you getting the idea? Understanding the idea? Though the parent contains two allele during gamete formation, the factors or allele pair segregate from each other. Now question arises, how it become possible? How it become possible? It become possible due to this reason. It is tall. It is tall. F1 selfing we are doing. So one small t present with this capital T, another small t present with this. And in next generation, who is coming? Dwarf is coming. Dwarf is coming means who is there? Small t, small t. So small t, how it is possible? When these small t separate from these capital T and when these small t separate from these capital T. So it is showing about what? Gamete are segregating. If gamete not segregate, if capital T, small t not segregate from each other, which combination is never possible? A small t combination is never possible, and this is about which one? Law of segregation of gamete. Now we are going to talk about incomplete dominance. See here what NCRT wrote about incomplete dominance. Have a look. So NCERT writing about incomplete dominance, कि when we make a cross between a red color flower of snapdragon or mirabilis, anyone, with a white color flower, we are getting F1 all pink color, means F1 not resemble with either parent. Not resemble with which one? Either parent. Not matching with any parent. When? So it is showing F1 showing which one? Phenotypic blending, not genotypic blending. It is showing phenotypic blending. What happened? Pink is the mixture of which one? Red and white. It is intermediate between red and white. Are you getting the idea? Pink is intermediate between red and white. But when we make the selfing of two pink, for which one? Pink. For which generation? F2 generation. We are getting phenotypic ratio in ratio of which one? 1 to 1, and genotypic ratio also in ratio of which one? 1 to 1. What it is showing? It is showing that genotypic blending not occur. If genotypic blending not occur, then only we are recovering which one? Red and white. Otherwise, red and white not come in the F2 generation. So only phenotypic blending occur, genotypic blending not occur. And another important is, in incomplete dominant in F2 generation, both phenotypic and genotypic ratio in the format of which one? 1:2:1. Got the idea? Now the third one is which one? Concept of dominant. Concept of dominance. So concept of dominant, going to say, let me say in a very short frame of time about the concept of dominance. I'm trying to fix it in 2-3 minutes. So concept of dominance saying, suppose that capital W is violet. Capital W, small w is violet. Means violet expressed in both homozygous and heterozygous condition. Small w is white. So why dominant is expressing in which one? Heterozygous and homozygous character both, and recessive only in which one? Homozygous condition. How it is happening like this? So for that, it is important for us to understand the expression of a gene. So capital W is a gene here. How it is expressing? It is making a mRNA by transcription. mRNA making a protein by translation. Suppose that this protein is an enzyme, E1 enzyme. This converting a raw material, it is a substrate, into a product pigment. Which pigment? Violet pigment. And it is a product. So how violet is controlled by capital W, did you understand? Capital W make a protein, this protein act as an enzyme, this enzyme catalyze a reaction, this reaction produce violet pigment. So any gene perform any function by protein synthesis. Whenever a work done by a gene, they perform by which one? Protein synthesis. So capital W making a violet pigment in such a way. Now what happened? This capital W undergoes in mutation. Mutation. So capital W is a which kind of allele? Unmodified allele. I am telling because here student struggle somehow. Or it is also called wild type. It is also called wild type. So it is converted into my small w. It is a modified allele. Or modified allele is also called mutant type. So when a small w forming, there are three possibilities are there. First possibility, they are producing same enzyme. And somehow it may be less functional enzyme. Possibility number 2 is they are producing non-functional enzyme. And third is no enzyme. Second possibility, third possibility. So when first possibility occur, any phenotypic changes occur, they are producing E1, it is producing E2, assume. So in first case, what happened? Modified allele is equal to non-modified allele. How can we gauge mutation occur or not occur? Mutation occur, but phenotype not changing. Same enzyme is producing. How can we say that mutation occur by observing it? So we can't consider the first condition. Why we are not considering first condition? Because first condition not giving any visual changes. Any visual changes. Before mutation, same phenotype. After mutation, same phenotype. Visual changes not occurring now. Contrasting character is not forming by which condition? First condition. So we will consider second and third condition. You forget first condition. What the idea? Because these mutation not give any change. Now suppose that capital W is there. Who is producing? E1 is producing, agree? If E1 is producing, raw material will converted into which one? Violet. Simply I am writing white. If capital W, small w is there, E1 will produce, yes. E1 will produce. Here also raw material converted into pigment, violet. Now if a small w is there, we will consider second and third, not first condition. Either E2, non-functional enzyme or no enzyme is producing. It will not convert raw material into which pigment? Violet pigment. So what we are finding, can I write a line? Can I write a line? Expression of a phenotype is due to presence of dominant allele, presence or absence of a dominant allele. If capital W is coming, violet is forming. If capital W is not there, who is forming? White is forming. Here no pigment is there, na? White is there. So who is responsible for expression? If capital W when present, who will form? Violet will form. When capital W absent, who will form? White will form. Who have no role in expression of the phenotype? Small w. Right? So capital W when present, violet, when absent, white. Because no any reaction occur, no pigment will produce. A small w not did anything here. When no work occur, white came. Understand the idea? So expression of phenotype depend upon presence or absence of a dominant phenotype or dominant trait. If a dominant trait is present, X phenotype will present, and if a dominant trait is absent, Y phenotype will come. So this is about the which one concept of dominance. Now we are moving ahead. Have a look here. Now we are going to talk about which one? Co-dominant. Beautiful example of the co-dominant is which one? AB blood group. Which one? AB blood group. Right. So if we talk about AB blood group, ABO blood group, I am writing ABO blood group. Blood group. So all have how many different alleles? It is 3 different alleles there. 3 different allele it has. So it is also example of which one? Example of multiple allele also. Multiple allele also. ABO blood group example of multiple allele also. Example of co-dominant also. And it is also example of dominant. So it is example of all 3. Dominant, co-dominant and multiple allele. How? Let me tell you. I gene is there. These I gene have 3 different allele. IA, IB and IO. So these all 3 are same gene but different allele. So I gene have 3 different allele, IA, IB and IO. What the idea? Now what happening? These IA, if you talk about IA, it is forming antigen A. Just let me write. Here it is writing now. Okay. If you are going to talk about which one? IA, it is responsible for antigen A. IB, it is responsible for, wait a minute. I am doing somehow hurry, so wait a minute. So 3 allele are there. But in an individual maximum, how many allele will come? 2 allele will come. So if we talk about multiple allele reported in population, why I am telling reported in population? Suppose that I am talking my ninth chromosome. This is ninth chromosome. Why I am telling ninth chromosome? Because gene for ABO blood group present in which one? Ninth chromosome. So this is my ninth chromosome. And assume my blood group is AB. Just assume. So what possibility is there? From one parent it came IA, another parent it came which one? IB. Understanding the idea? So out of 3 allele, how many allele present in my body? Only 2 allele. So if you want to study about O allele, you need one more person. Na? In an individual, maximum 2 allele of a gene are present. So that's the reason we are telling multiple allele are reported in which one? Population. Are you getting the idea? Is it clear in the mind? So for another O blood group, I need one more person. Na? Got the idea? So in an individual, maximum 2 copies there. So what the possibility is there? 3 allele combination. So some person have IA. Some person have which one? IA, IO. Some person have which one? IA, IB. Means one parent I, another parent IB. Some person have which one? IB. Some person have which one? 1, 2, 3, 4. IB, IO. Some person have which one? IO. Both parent contribute. So 1, 2, 3, 4, 5, 6. 6 possible combination is there. So A, here it is going for A blood group. A is dominant over O, so it is A blood group. AB are co-dominant, so it is AB blood group. B is here B is dominant over O, here O blood group. So here it is phenotype. Here it is which one? Genotype. So how many genotype we find for the human ABO blood group? 6 genotype. And how many phenotype is there? 4 phenotype. Got the idea? Is it clear? So what happening here basically, IA antigen is responsible for a reaction and which is going to make a which one? Carbohydrate. So in A antigen, what you are finding? Which carbohydrate is attached? NAG is attached. In B antigen, who is attached? Which carbohydrate is attached? Galactose is attached. And in which one? O blood group, no antigen is there on the surface of RBC. No antigen on there in the surface RBC. So if anybody have A blood group, which antigen is present on surface of RBC? A, which carbohydrate is attached here? NAG here, galactose here, no antigens is there. Got the idea? Is it the reason NCERT wrote here something regarding the sugar? What they writing? The allele IA and IB produce a slightly different form of a sugar. NCERT not wrote the name the sugar, and a small I, IO is also called which one? A small I not produce any sugar. So here no antigen is there. This sugar acting as a which one? Antigen. Are you getting the idea? Attached with the protein. Got the idea? So NAG is there, galactose is there, and here who is there? No antigen is there. So these are about ABO blood group. Now examiner asked some question from here. What question asked? Number of genotype in multiple allele equal to n into n plus 1 upon 2. In human blood group, how many allele is there? 3. So 3 into 3 plus 1 upon 2, 3 into 4 upon 2, 3, 2 2s are 4, 3 2s are 6. How many different genotype we have seen there? 6 different genotype we find now. So question, people will ask some numerical nature of question from which one? Multiple allele. Now ABO blood group, AB blood group is example of which one? Co-dominant. Suppose that a female is AB blood group. She is married with a male with AB blood group. What are the possible blood group in children? Both male and female are of AB blood group. What are the possible blood group in children? So what we will do? How to handle? You can handle like Mendel also and normal also. I am writing IA, IB. So here it becomes which one? IA, IB, IB, IA, Here IB, IB. So here A blood group, AB blood group, AB blood group, B blood group. So what is the phenotypic ratio you are finding? Phenotypic ratio 1 to 1. Genotypic ratio what you are finding? 1 to 1. Why? Because this is 1, this is 2, this is 1. So in codominant also we are getting what? Same phenotypic and genotypic ratio. When we are selfing the same genotype of heterozygous type. Got the idea? It is heterozygous condition. Huh? Got the idea? So here we are getting the same kind of result. Now we are going to talk about further what NCERT wrote, dihybrid cross and independent assortment. Suppose that I am making a cross between a round and yellow, with round yellow with wrinkled green. Parent of F2. So capital R, capital Y cross with a small r, small y. What is the gamete came? Capital R, capital Y. Here who will come? A small r, small y. What happening here? Capital R, capital Y, small y. Who is there? F1 is there. Okay. All will be which one? Round and yellow. All will be which one? Round and yellow. Now we are selfing the F1 for F2 generation. Capital R, capital Y, capital R, capital Y, small y. What happening here? 4 type of result. Capital R, capital Y, capital R, small y, capital Y, small y. You are making Punnett square. Here capital R, capital Y, capital R, small y, capital Y, small y. What phenotypic ratio you are getting? 9:3:3:1. What genotypic ratio you are getting? 1:2:1, 2:4:2, and 1:2:1. So dihybrid is nothing. It is the multiplication of monohybrid. No need to do this cross. Everyone you can break in the monohybrid. Just look here. 3:1, one monohybrid into 3:1, another monohybrid. Mono, dihybrid. So it becomes 9:3:3:1. So you multiply 2 times monohybrid, dihybrid result will come. Genotypic ratio multiply 2 times monohybrid genotypic ratio. Whose ratio will come? Dihybrid ratio will come. Got the idea? 1:2:4:2:1:1. Oh, 1:2:1. I am in too much hurry. 1:2:1, 2:4:2, 1:2:1. This is the ratio came. If you multiply it, same ratio came or not came. So you simply multiply the things. Now I am telling, based on dihybrid cross, Mendel proposed independent assortment. How independent assortment? Look here. In F2 generation, Mendel get 3:3, 9:3:3:1. It is round and yellow. It is important you understand here. It is which one? Round and green. It is which one? Wrinkled and yellow. It is which one? Wrinkled and green. Did you got the idea? So what here Mendel find? It is parental, it is non-parental, it is non-parental, it is parental. So non-parental phenotype is only possible. You just look here now. You see the parent. In parent, who is together? Round and yellow is together. Wrinkled and green are together. But in children, F2 generation, round and green is also coming. Means round and yellow are not linked together. They are independent from each other. If it is independent, then only round going with which one? Green. So there is no commitment between which one? Round and yellow. And even there is no commitment between which one? Wrinkled is green. Wrinkled going with green also, wrinkled going with which one? Yellow also. Round going with yellow also, round going with which one? Green also. So they are not linked together and they are independent from each other. They are independent inheritance is independent. So 9:3:3:1 ratio in F2 generation, and recombinant phenotype, it came na 6:16 recombinant phenotype or non-parental phenotype, recombinant or non-parental, and 10:16 came which one? Parental. It is due to which one? Independent assortment. It is due to which one? Independent assortment. Now how to handle the question based on that? How to handle the question based on that? I am asking a question. I am asking a question. Capital A small a, capital V small v cross with capital A small a, capital V small v. Question is, in which ratio we obtain a small a, capital V small v, capital A small a, capital B small v, capital A small a, capital B, capital A small a, small v, another small a, capital B in their progeny. So here you look, we are making the selfing of F1. If we are going for making the selfing of F1 heterozygous, what is the chances of this? So there is a trick for it. What is the trick for it? Number of a genotype in F2 generation is equal to gamete produced by that genotype. So here small a, capital V is there. How many gamete produced by it? 2 to the power n, you know na? It is forming 2 type of gamete. So 2/16. Punnett square is how much? Dihybrid cross 16. Here how many type of gamete produced by it? 4/16. 16 is what? Punnett square I am writing. Here it is how much type of gamete? 2/16. Here what is the gamete produced by it? 1/16. So what is the ratio came? Ratio is 2:4:2:1. If you want to denominator, write the total number of Punnett square. Do one more question. I am making a cross, capital A small a, capital B small v, capital C, capital D small d, capital E small e, cross with small a, capital V small v, capital C, small d, capital E small e. In their children, what is the chances of getting a small a, capital B small v, capital C, small d, capital E small e? We are making the cross between these two. What is the chances of getting this? We will not use that tricks because it is not the selfing of heterozygous. It is not all heterozygous. Na? Are you getting the idea? Even the genotype of the two parents are not same. So how to handle this question? Break everyone in monohybrid and multiply it. How? Make the cross between this and this. Tell me the chances of this. I am doing for one and then I will write in a speed. Capital A small a, small a. Capital A small a, here it is one type of gamete. What will come? Capital A small a. What is the chances of getting a small a? 1 out of 2. So 1/2. Into capital V small v, chances of getting this one, 2/4. Capital C, small c, chances of getting capital C, small c, 1/2. Capital D, small d, chances of getting a small d, 1/2. Capital E, small e, chances of getting this is 2/4. So what the answer came? You can, if you wish, you can cancel it in the smallest form also. So multiplication become easy. It is 1/2. It is 1/2. What answer came? 1/2. 2 2 4 2 8 2 16 2 32. Answer. So you break everyone in which one? Monohybrid, multiply it, answer will come. Got the idea? Okay, right? Now we are moving ahead on the NCERT. So I am ready to teach if you are ready to listen. Okay, so have a look, give your attention. Now we are going to talk about this side. Chromosomal theory of inheritance. Give your attention. Chromosomal theory of inheritance. So NCERT wrote first कि Mendel proposed his idea in which year? 1865. Presented it, published it. But Mendel not recognized overnight. Right? There are many reasons behind Mendel not got the instant recognition. The first is what? He published his finding in a small journal. Media was not aggressive that time. So many people not able to understand, know any such kind of research paper came. Got the idea? Other reason was also there. Mendel was the first person use what? Mathematics first time in the biology. Even their contemporary not able to understand what he want to say by mathematical language. Third reason is what? Mendel. Third reason is what basically? Mendel not given any proof for which one? Factor. When people ask what is factor, he told something. And fourth reason was what? People was not accepted the Mendel idea, factors moving as a discrete unit stably to the next generation. Because in natural population variation are there, but Mendel not explain the variations. And that is the reason Mendel idea not become so popular at his time, and Mendel died unrecognized in 1884. But in 1900, three different scientists from three different countries, Hugo de Vries, Sermark and Karl Correns, reached on same conclusion which already established by Mendel 35 years ago. Got the idea? And once again, rebirth of the Mendelism occur. Once again, rebirth of the Mendelism occur. So as the rebirth of the Mendelism occur, entire scientific community struggle to find what is factor. And in just 2 years, Sutton and Boveri claimed that Mendelian factor is chromosome. Or he told chromosome moving from one generation to another generation. What is the reason behind their claim? The reason behind their claim, they told both gene and chromosome occur in pair. The reason behind their claim was both gene and chromosome segregate in such a way out of a pair only one is passing in a gamete. And they also told both independent pair of gene or chromosome inherit independently. What the idea? So somehow they are right, but they are not exactly right. Actually, chromosome is not gene. Gene is present on chromosome. So, but there are so many parallelism they established between gene and chromosome and able to convince the world that is what? Chromosome moving from one generation to another generation. Now here student confuse A side or B side. So A side is gene, B side is gene, A side is chromosome, B side is not, sorry, A side is gene, what I am telling. Wait a minute. A side is gene, let me write. What happening? A side is gene, B side not a gene. Wait a minute. What happened? Just give me a moment. A side is, why is not writing? A side is, it is lagging, probably. Okay, let me say, it is not working, it is probably lagging. A side, all point of A side is proved by gene. All point of A side proved by chromosome. All side of B is proved by chromosome, but all side is B not proved by gene. So who never be a gene? Here B. Why? Because of third point. One pair of gene segregate independent to other pair of gene, it is always not true. Gene may show linkage, but chromosome never show linkage. Gene may show linkage, but chromosome never show linkage. So it is gene, it is chromosome, it is not gene. That I want to say. Every time I want to write this one. Many times we ask question and many people do the mistake here. That's the reason I want to write. So this is what? Gene also plus who is also? Chromosome also. This side only which one? Chromosome. Prove only chromosome, not gene. Reason? This point. One pair segregate independent to other pair, always not true for gene. Board is also feeling hungry, I hope. Not true for gene. Got the idea? Two pair of gene may show what? Linkage. If it is present on same chromosome and they are close to each other. Now we are going to talk about Morgan study, Morgan inheritance study. It is what happening, God knows. Now give your attention, cancel. Now we are going to talk about T.H. Morgan. So if we talk about the term linkage and recombination is coined by which one? T.H. Morgan. T.H. Morgan performed so many experiments by taking Drosophila melanogaster. Performed so many experiments by taking which one? Drosophila melanogaster. Getting the things or not? So here two experiment is given in NCERT. In one experiment, he make a cross between a female Drosophila with a yellow body and white eye with wild type. Wild type means brown body and red eye. In F1, he find crisscross inheritance occur. Got the idea? So female child become like father and male child become like mother. Got the idea? When he make the cross between these F1 progeny, in F2 generation he find 98.7% progeny is like parent and recombinant is how much? Only 1.3%. When he make a cross between another character of the Drosophila, that is white eye and miniature wing, with wild type means which one? Red eye and long wing. Red eye and long wing. In F1 here also they find crisscross inheritance occur. And he make the cross between F1. In F2 generation he got 62.8% parental and 7.2% recombinant. Question arises. Here we got the different result and it is experiment result. It is not a mathematical result. What he got in the experiment, he calculated all Drosophila and write the result in the percentage. Morgan told these are the result due to linkage. And why there is a differential result? The reason of differential result is told keep parental character depend upon the distance between two genes. If two genes are close to each other, the chances of parental outcome is more. And if two genes are far on a chromosome, the chances of parental outcome is relatively less. But in case of linkage, parental is always greater than recombinant. In case of linkage, parental is always which one? Greater than which result? Recombinant result. And he make a conclusion like which one? Ki linkage based on this result is inversely proportional to which one? Distance between two genes on a chromosome. Two genes on a chromosome. Got the idea? So you understand about this experiment of which one? T.S. Morgan. Now one of the student of Morgan, what is the name? Alfred Stewart Went. Alfred Stewart Went discovered which one? Gene mapping. They developed which one? Gene mapping. And how we determine the distance between two genes? By recombinant frequency or crossover value. What is the formula for recombinant frequency? Recombinant frequency equal to total number of recombinant divided by total number of progeny multiplied by 100. So suppose that an example recombinant frequency came 1%. So distance between two genes is which one? 1 map unit. And Got the idea? So it is not an actual distance between two genes. It is a assumed value. Ki how much percent recombinant frequency will come, the distance between two genes become that much map unit. Are you getting the idea? Now if we go for two things are there, pleiotropy and polygenic in this chapter. Let's come on this. So we are going to talk about polygenic inheritance. Polygenic inheritance. What happened in polygenic inheritance? This board is not supporting. Let me write only. Leave it. Leave NCRT. It is somehow creating problem. Wait. Okay, let me speak. Nothing is happening. So let me speak about polygenic inheritance. Leave it now. Polygenic inheritance. So polygenic inheritance is a kind of quantitative inheritance. Quantitative inheritance. What happens in quantitative inheritance? When more than one gene control one character is called polygenic inheritance. More than one gene responsible for one character. More than one different gene. Or better to write somewhere people writing, which got the idea. So an example, human skin color controlled by 3 genes. Got the idea? So almost character who are in a population who have a gradient of phenotype is controlled by polygene. You see your class, height of the different people are different, IQ of different people, no two range is there. It's not like tall and dwarf, two end is not there. Between tall and dwarf, many gradient is there. So all the character who have a gradient in a population controlled by polygene. An example is what? Human skin color, human height, human IQ, human eye color. Got the idea? These all are governed by which one? Polygene. Got the idea? So if we talk about the polygenic inheritance, what is important things is there? Due to that, character present in a population in a gradient. And result of the polygenic inheritance, if you draw a graph of the phenotype in a population, you got a bell-shaped graph. So you see your class, most of the people have an average height. Okay? Too extreme is less. You see your class, you will find most of the people have which kind of a skin color? Intermediate, mulatto skin color. What you are saying? Chamal chaya. So dark is less, white is less, but intermediate skin color is more. So what happened? If we talk about phenotypic ratio in F2 generation of a polygenic inheritance of human skin color, what phenotypic ratio is coming? If it is working, I can write. What phenotypic ratio is coming? 1:6:15:20:15:6:1. Got the idea? So this is the phenotypic ratio of which one? A skin color. And any character where polygene, how many polygene? 3 polygene is responsible. When 2 polygene is responsible, what is the phenotypic ratio in F2 generation? 1:4:6:4:1. If I draw a graph of both phenotype, we are getting a which kind of graph? Bell shape of graph. Are you getting the idea? Another thing I am telling you, that is what? Number of phenotype in polygenic inheritance equal to number of phenotype in polygenic inheritance equal to 2n plus 1. So in human skin color, 7 phenotype is there. How many genes are there? 2 into 3 plus 1 equal to 7 for skin color. Now pleiotropy gene. Pleiotropy gene is what basically? Pleiotropy gene is what basically? When one gene control more than one character. Such kind of gene is called what? Basically pleiotropic gene. Are you getting the idea? When one gene control more than one character, it is called what? Pleiotropy gene. Got the idea? So gene for phenylketonuria is an example of pleiotropy gene. Actually, what happened in phenylketonuria? Phenylalanine is not able to convert into tyrosine. So generally, phenylalanine converted into tyrosine via enzyme phenylalanine hydroxylase. So when this gene who is responsible for this enzyme become defective, this enzyme will not produce. If this enzyme will not produce, these phenylalanine not convert into tyrosine, who accumulate into the body? Phenylalanine accumulate into the body, it accumulate in cerebrospinal fluid and it causes some symptom, and symptom are mental retardation. Symptom is reduced pigmentation. Got the idea? In which one? Hair and skin. And another example is what? Short stature. So one gene is affected, but more than one phenotype it is showing. So it is an example of which one? Pleiotropic gene. Another example of pleiotropic gene is which one? Even gene for sickle cell anemia. In sickle cell anemia, so many symptoms came. So when any diseases where due to any gene whose defect causing many symptoms, that gene is working as a which one? Pleiotropic gene. Got the idea? So this is about the theoretical aspects of the principle of inheritance. Got the idea? Right? So numerical you can practice, because in this session if we just work on numerical, nothing theoretical touching will possible. Now are you comfortable? Can I move on reproduction? So look here, 15 minutes if it is possible. Can you? Are you ready? At least finish the second year topic. 15 minutes I will take. Sexual reproduction. Doubt you ask later. Oh, what to do? NCERT is not dragging. Okay, I am speaking. Okay. It's dragging too slow. What happening, bhaiya? Leave it. So I'm just telling fastly about microsporogenesis and megasporogenesis. Look here. Here it is stamen, here it is anther. It is bilobed structure. What the idea? When we are cutting the TS of anther, we are getting such kind of a structure. So each lobe have two chamber, dithecous, and each chamber is what? Basically microsporangium. Each chamber is microsporangium. So a typical anther is a dithecous, bilobed, tetrasporangiate. Got the idea? If we talk about microsporangium, it is covered by which one? Anther wall. So if you talk about anther wall, I am not even making epidermis. Below that, who is there? Endothecium. Then who is there? Middle layer. Then who is there? Tapetum. What idea? These are the wall of anther. So out of the 3 layer of the anther wall, these 4 layer, these 3 are protective in function. Epidermis, endothecium and middle layer. And tapetum provide nourishment. Inside of the microsporangium, what we are finding? Inside this microsporangium, who is there? Sporogenous tissue is filled. sporogenous tissue. And this is what basically microsporangium. Microsporangium. Later these microsporangium called pollen chamber or pollen sac. So they are filled with a sporogenous tissue. Each cell of a sporogenous tissue is a potential microspore mother cell. Microspore mother cell. Each cell is a potential microspore mother cell. Are you getting the idea or not? Now what happened here? This is microspore mother cell, MMC. It is going for meiosis 1. It is going for which one? Meiosis 2. And it is forming microspore tetrad. Microspore tetrad. When they dehydrated, it separated and it is what? Microspore. Microspore. Sometimes people also told these microspore as immature pollen grain. Got the idea? Now what happened? These microspores divide by mitosis. Got the idea? Two cells are forming. One is vegetative cell. Another is generative cell. If you talk about in more than 60% of angiosperm, pollination occur in which one? 2-cell stage. In remaining, it occur in 3-cell stage. So what happened later on where 3-cell stage pollination occur? Who also divide? Generative cell divide by which one? Mitosis, and it is forming 2 male gamete. Ultimately, so it is converting into 3-cell stage. Got the idea? So if you talk about a mature pollen grain have a vegetative cell and a generative cells. Isn't it clear in the mind? That line written in NCERT. But finally it is what? Converted into which one? 1 vegetative cell and 2 male gamete. Further, who also divide by mitosis? Generative cell divide by mitosis and forming male gamete. So this is about the formation of a pollen grain. Got the idea? Now if we talk about, so it is which one? Male gametophyte. Or pollen grain. Mature pollen grain. Isn't it? Later on, if we talk about pollen grain surrounded by exine and intine. If we talk about exine, it is made up of a sporopollenin. It is one of the material who is highly resistant, not degradable by any heat, cold and any kind of enzyme. And due to a sporopollenin, we can making the fossils. We are finding the fossils evidences of the pollen as well. Got the idea? Where pollen exine is not there, sporopollenin is not there, pollen tube is there. Intine is made up of pectocellulosic material. Now we are going to talk about which one? Female side. So here you look just where I'm just going to draw a brief diagram. Here it is stigma. It is somehow flask shape, structure, style, and it is ovary. And it is thalamus where it is present. Now inside the ovary, who is there? Ovule is there. This is ovule present. Got the idea? So you just see here, it is what? Funicle of the ovule. And it is which one? Placenta. Got the idea? So if we talk about a ovule, a typical ovule is also called which one? Mega sporangium. So in a typical ovule, what we are finding, it is the integument of the ovule. It is outer integument, inner integument. This is funicle, stalk of the. It is which one? Where funicle attached with which one? Body of the ovule. It is which one? Hilum. Are you getting the idea? This is what basically chalaza. This is what basically where integument is not there, micropyle. And here it is outer integument. Here it is inner integument. Now inside the ovule, who is present? Inside the ovule, inside of the ovule, you can say nucellus is present. What is the ploidy of the nucellus? 2n. Got the idea? Now what happened? One of the nucellar cell who is present toward which end? Micropylar end. One of the nucellar cell which is present at which end? Micropylar end. We have as which one? Megaspore mother cell. Megaspore mother cell. Ploidy is 2n. These megaspore mother cell divide by meiosis 1 and meiosis 2. 4 daughter cell produce out of 3 in most of the plant. 3 megaspore degenerate and only 1 functional megaspore is there. Only 1 functional megaspore is present. Got the idea? Now if we talk about these only 1 functional megaspore, what happened? These only 1 functional megaspore is there. These functional megaspore going for monosporic germination. Which germination? Single megaspore forming the embryo sac. They are going for free nuclear division from 1 nuclei to 2 nuclei, from further mitosis 2 nuclei to 4 nuclei, further mitosis but only karyokinesis 4 nuclei to 8 nuclei, free nuclear division mitosis. Now these 8 nuclei reassemble like this. Here I am telling which one? Micropylar end, chalazal end, and here I am writing micropylar end. Reference to anatropous ovule. So out of 4 toward micropylar end, 4 toward chalazal end. Then further it will rearrange. 3 remain here, 2 come in center, 1 somehow we come forward and arrange like this. And then cytokinesis occur. So these 3 cell structure present at the micropylar end is called egg apparatus. Egg apparatus. These 3 are antipodal cell, and this bigger cell is called central cell, and in central cell these 2 are what? Polar nuclei. So this structure is called embryo sac or female gametophyte or female gametophyte. This embryo sac and female gametophyte is a 7 cell 8 nucleus structure. And only how many cell have cell wall? Only 6 cell have cell wall. Central cell don't have cell wall. So in such a way, formation of which one? Female. Now we are going to talk about pollination. You better know autogamy, when pollen can transfer from anther to stigma of the same flower. No agents required. Got the idea? Now we are going to talk about gaetenogamy, when pollen grain transfer from anther to stigma of a different flower of the same plant. It is genetically cell pollination but functionally cross pollination. Pollinator required in gaetenogamy. And genogamy in true sense cross pollination, when pollen grain transfer from anther to stigma of the which one? Different plant. Got the idea? That is genogamy. What is clisteogamy? Closed flower. What is chasmogamy? Open flower. In clisteogamy only who will occur? Autogamy occur. Merit of clisteogamy: pollination is certain, seed formation is certain. Demerit is inbreeding depression. Chasmogamous flower. Got the idea? Clisteogamous flower means autogamy. What is the demerit of autogamy? Demerits is what? Always cross-pollination, self-pollination occur because closed flower is there. Understand? Now we are going to talk about chasmogamy. Chasmogamy means open flower. In open flower, both self and cross-pollination are possible. Any plant, can you take the example where both clisteo and chasmogamy is here? Yes. What are the name? Oxalis, Commelina, Viola. These are the some of the plant who have which one? Both closed as well as open flower. Now we talking about the agent of pollination, abiotic and biotic. In abiotic agent, wind is a more common abiotic agent than water. Got the idea? Water is a rare kind of which one? Abiotic agent. Now we are going to talk about abiotic agent, wind pollination. What are the character present? So pollen grain lightweight, pollen grain non-sticky, expose male and female reproductive organ. Stigma is feathery. Got the idea? Pollen grain blow in the air up to the long distance. Pollen grain produce in large amount. These are some of the adaptation present in which one? Air pollinating flower. Right. There is no need of nectar. There is no need of scent in air pollinating flower. Got the idea? And important is what? Ovary have single ovule in wind pollinating flower. NCERT wrote na? You see the diagram of the which one? Corn. So corn, cob in maize is nothing other than stigma and style. So wind pollination done. Water pollination. What is about the water pollination? So if we talk about the water pollination, NCERT wrote कि in lower plant water is essential for fertilization to occur. Like in algae, bryophyta, pteridophyte, water is essential for fertilization. But if we go for the higher plant like angiosperm, dependency on water for reproduction reduce up to which one? 30 genera only, and out of that mostly of which one? Monocot. Even all the plant who are aquatic not get pollinated by water. An example given is which one? Water lily, and water hyacinth. These are the some of the plant where pollination not occur by water because their flower is exposed over the surface of water. So they get either pollinated by wind and insect. Now, example of hydrophily is of two type, epihydrophily and hypohydrophily. What is the example of epihydrophily? NCERT को give? Vallisneria, hydrilla, it is an example of epihydrophily, and Zostera, that is a seaweeds. Here pollination occur under the water, that is the example of hypohydrophily. Where pollination occur by water, pollen grain have a mucus coating, means it is remaining dry. It is a hydrophobic coating. So it is preventing the pollen grain from getting dry. Now we are going to talk about biotic agent as a pollinator. It is called zoophily. When water hydrophily, wind anemophily, and biotic agent zoophily. Zoophily is of many types. If insect is a pollinator, entomophily. If bat is a pollinator, chiropterophily. If a snail is a pollinator, malicophily. So many different animals, even primate, non-primate, rodent act as a pollinator. Got the idea? Bird act as a pollinator, ornithophily. Got the idea? So many different type of zoophily is there. So what are the adaptation of entomophily? Flower have nectar. Flower have which one? Bright coloration. Right. These are the some of the floral reward to attract the which one? Pollinators. Getting the things or not? So if you talk about in some plant, what happened? Pollination also occur during finding the safe place for laying egg. NCERT wrote two example, Amorphophilus and Yucca. Yucca only pollinated by Yucca moth, and it is an example of coexistence also. If Yucca will die, Yucca moth will die. Later on textbook writing about which one? Pollen robber. Pollen robber are that organism, that animal who visit the flower and take the nectar but don't pollinate them. Now we are going to talk about which one? Outbreeding devices. How many outbreeding devices means what? That devices who favor cross pollination and retard self pollination. Got the idea? Favor cross pollination and retard self pollination is called outbreeding devices. What are the name? Unisexuality. If a plant is unisexual, dioecious, only who will occur? Cross pollination occur. Got the idea? Now you can say about no synchronization in maturation of male and female reproductive organ. Dichogamy we are telling it. So protandry and protogyny is a outbreeding device. It will support whom? Cross pollination. It will not support whom? Self pollination. Other outbreeding device is which one? Hercogamy. Anatomical barrier is there. Stigma is this side, anther is this side. How they will going to? Got the idea? So this is hercogamy. And self incompatibility. What happen in self incompatibility? If the pollen grain of same genotype landing on the stigma, what happened? They not germinate. Pollen pistil interaction not occur. They reject them. So nature also in the support of which one? Cross pollination. And how they making it facilitate? With the help of outbreeding devices. Outbreeding devices for support of cross pollination, it is not supporting which one? Self pollination. Now, pollen pistil interaction. If we talk about pollen pistil interaction, all the event starting from landing of pollen grain on the stigma to pollen germination and entry of pollen tube in the ovule is called pollen pistil interaction. Got the idea? So who is guiding the which one? Pollen tube during pollen pistil interaction? Filiform apparatus. It is attached with the synergid. They are guiding the pollen tube, and pollen tube enter into the embryo sac only through which one? Micropylarine, and that is called porogamy. NCERT only talk about porogamy. Got the idea? Now what we are going to talk about? Double fertilization. What happened in double fertilization? So egg cell fertilized with one male gamete, egg cell plus male gamete making zygote. This event is called syngamy. And polar nuclei, it is polar nuclei, PN, another polar nuclei. It is fusing together and making N. Wait a minute. Here making 2N structure, it is called secondary nuclei. So secondary nuclei have which ploidy? 2N ploidy. How secondary nuclei fuse? When two polar nuclei fuse together, and when male gamete is coming, it is forming which structure? 3N structure, that is called primary endosperm nuclei. So this event is called, 3 nuclei is fusing, this event is called triple fusion. And it is called syngamy. Together they are called double fertilization. So why I told separately, secondary nuclei have 2N ploidy, polar nuclei have N ploidy. Zygote have of course 2N ploidy. So after post fertilization, what happened? First, primary endosperm nuclei divide and form endosperm. So endosperm when also forming, first they are going for free nuclear mitosis, then they are going for cellular mitosis. So NCERT wrote coconut, tender coconut, you are getting both. Coconut water have which one? Free nuclear stem, endosperm, and coconut white meat have which one? Cellular endosperm. So these endosperm provide nourishment to develop zygote into embryo. When zygote develop into embryo, what happen? Zygote first convert into which one? Pro-embryo, then it is forming into globular embryo, then it is forming into heart-shaped embryo, getting the thing, then it is forming into mature embryo. If you talk about a mature embryo of a dicot, it have two cotyledon and embryonal axis. Cotyledon, embryonal, which one? These are two cotyledon and this is embryonal axis. Embryonal axis above the cotyledon is called epicotyle. Below of it is called which one? Hypocotyle. Hypocotyle. Got the idea? Here tip of the epicotyle is which one? Plumule. And tip of the hypocotyle is what? Radical. Radical. Are you getting the idea? Plumule gives to shoot system, radical give to root system. In monocot embryo, how it is different from the dicot embryo? If NCERT diagram is opening, it is better for me, not required to draw it because diagram is somehow complicated. Wait a minute, if it is opening, let's see. Finder, let me open it. Must level the every diagram of the NCERT. Just let me talk about the dicot embryo where is their sexual reproduction. Conditional, it open. You open your NCERT. If it is not drag, I will say you just, no, nothing is happening. It is not scrolling. Lag is coming, no problem. You open your NCERT. So out of two cotyledon, monocot embryo you see in the diagram, NCERT diagram, out of two cotyledon, got the idea? Out of two cotyledon, one cotyledon become rudimentary. That is called what? Epiblast. Rudimentary cotyledon. Now one functional cotyledon are there that are lateral in position and shield shape. That is called which one? Escutellum. Escutellum is the functional cotyledon. Got the idea? Escutellum is which one? Functional cotyledon, and rudimentary cotyledon you have seen now. Later on what we are finding? Plumule is covered by which one? Coleoptile, which is a hollow structure. Plumule is covered by which one? Coleoptile. And radical is surrounded by a shield-like structure that is called which one? Coleorhiza. That is which one? Coleorhiza. Understanding the idea or not? Okay. So presence of escutellum, presence of epiblast, presence of presence of coleoptile, presence of coleorhiza is a character of monocot embryo. It is not present in dicot embryo. So embryo is over. Got the idea? Now we are going to talk about which one? Seed, I am leaving it. Got the idea? Now I am telling you apomixis. Apomixis I am telling you. Have a look here. Apomixis. So what is apomixis? It is a kind of asexual reproduction. Which mimic sexual reproduction. How it is mimicking? Because seed is the final product of sexual reproduction. Na? So during apomixis, what happened? During apomixis, formation of seed occur without fertilization. Formation of seed occur without fertilization. Are you getting the idea? So some example is what basically family Asteraceae, member of Gramineae, member of Asteraceae have such kind of apomixis we are reporting. Got idea? So in apomixis, seed formation occur without fertilization. Now NCERT wrote about which one? Polyembryony. They are not told about the mechanism of it, so I am not telling here. So polyembryony. So they are writing in citrus and mango, there are more than one embryo is present inside a which one? Seed. And that is called which one? Polyembryony. Are you getting the idea? What is the advantage of apomixis? So NCERT wrote nowadays plant breeder are looking toward apomixis to avoid inbreeding depression. Actually what happened? When a scientist develop a hybrid seed variety, and when this hybrid seed variety undergoes in sexual reproduction, during sexual reproduction what happens? Genetic recombination occur. And when genetic recombination occur, what happens? Character is going to segregate. Are you getting the idea? So scientists are trying to develop these variety through which one? Apomixis. It is a kind of asexual reproduction. Na? So when we develop the hybrid variety through apomixis, no inbreeding depression occur. Because no meiosis occur, segregation not occur. Understanding the idea or not? So if we are developing any variety through apomixis, what happen? Never inbreeding depression occur of a hybrid variety. And that's the reason NCERT wrote in the second paragraph, the demerit of the sexual reproduction is what? It is segregating the gametes. What the idea? Segregating the character. So due to that reason, inbreeding depression occur. And if we talk about to avoid the inbreeding depression, who is the ray of apomixis? Now one student asking, sir, 1 ATP production, 4 proton. Yes, 4 proton. Right. Okay. So this is the apomixis. Morphology, anatomy, what to do? So second year, anyhow it is over. We can't expect in one day we are going to cover everything, but we tried our best to handle it. In biotechnology, we just talked like the things like very properly. I hope you understand. And molecular, we also talked about. So in principle of inheritance, you take care of the numerical. Reproduction, you read. Morphology, anatomy, see, we will, any other days, we will think about it if you have a time. And if you are interested, we can plan any day. Otherwise, you read it. Okay. So thank you very much. I hope you are also hungry. I am also somehow little tired. Got the idea? Thank you very much. Bye-bye. Number. It is in group. It is on group. Okay, ji. Then thank you.